First, I'll start by saying that I will be making some forward-looking statement. And I'm super excited because DNA is changing for the world for the better. That's what we do. We write DNA from scratch, and we want to help you solve the many critical challenges that humanity faces. The focus today is to focus on drug discovery and development, where synthetic DNA has the potential to rapidly drive improvement in our ability to treat diseases. So before we go into the details, a quick reminder that today the pharmaceutical industry is facing a real challenge where the cost of developing new drugs is now so high that the rate of return on investment is soon going to be negative. And to reverse this trend, we're going to need new modalities of drugs to increase the number of drugs. And we also will need to increase the efficacy with which we identify new drug targets, and to make sure that we're working on target that will have the highest likelihood of being successful in a very cost-intensive stage of clinical trials. So we need to increase the efficiency with which we identify safe and effective new drug against those targets. And finally, we need to increase the quality and expected commercial value of this drug. And hopefully that's where. Hi, my name is Emile Leproust. I'm the CEO of Twist Bioscience and one of the 3 co-founders. I'm very happy to talk to you about writing the future of drug discovery and development today. So first, I'll start by saying that I will be making some forward-looking statement. And I'm super excited because DNA is changing for the world for the better. That's what we do. We write DNA from scratch, and we want to help you solve the many critical challenges that humanity faces. The focus today is to focus on drug discovery and development, where synthetic DNA has the potential to rapidly drive improvement in our ability to treat diseases. So before we go into the details, a quick reminder that today the pharmaceutical industry is facing a real challenge where the cost of developing new drugs is now so high that the rate of return on investment is soon going to be negative. And to reverse this trend, we're going to need new modalities of drugs to increase the number of drugs. And we also will need to increase the efficacy with which we identify new drug targets, and to make sure that we're working on target that will have the highest likelihood of being successful in a very cost-intensive stage of clinical trials. So we need to increase the efficiency with which we identify safe and effective new drug against those targets. And finally, we need to increase the quality and expected commercial value of this drug. And hopefully that's where we can help. So how can Twist help address those challenges? Well, we believe that our platform that miniaturizes the chemistry of writing ACGT on our silicon chip, maximize the throughput. We can make millions of oligos every day, and lower the cost of DNA, which can now unlock a number of applications. So let's start with target identification. The last decade, I've seen some incredible breakthrough in our ability to discover novel targets for treating disease. And we can't talk about that target identification without talking about CRISPR and how genome-wide CRISPR screening has enabled finding genetic elements that are linked to phenotypes. And thanks to the millions of oligos that Twist makes, now very high quality data can be produced. And here in the example published in 2019 by the Sanger Institute in Cambridge, they've shown that screening with Twist oligo pools of 324 human cancer cells of 30 different types resulted in really high quality data and a number of candidates for downstream cancer therapeutic. But now scientists are able to move beyond just silencing one region by using dual guides. And that creates a bunch of new applications in functional genomic screening. For instance, you can excise promoters, you can excise link RNA, you can excise non-coding elements, or you can actually look at systematic interaction of one gene pair with another gene pair. And so the barrier to entry to be able to do that is actually the length of a dual guide. A dual guide is beyond the ability of normal DNA synthesis, but at Twist we have addressed that challenge by being able to make 300 base pair oligos. And so that means that now scientists can fit two single guide RNA together. They can fit a Cas12a with a CR-RNA array and basically expand the design space that they have. And so now we're enabling combinatorial screens without having to worry about the downstream cloning issues, without to worry about the cost. We can take care of all of it at a very cost-effective manner. And as an example, we are now seeing a lot of great publication in the literature using those dual guide scanning. So now that we've done the target identification, we can go into the drug discovery and development. And we believe that Twist has the potential to accelerate biopharmaceutical and immunotherapy development through rapid antibody optimization and humanization. And that's super important because in 2019, 4 out of the 5 best-selling drugs were biological. So that ability to discover and optimize biological drugs is key. However, it's inherently complex to optimize biological drugs, and the development takes time. And so at Twist, we offer scientists a toolbox to enable them to quickly find new and efficacious leads against targets or even against hard-to-drug target, which means, and then we give them the means to very quickly optimize drug-like properties for those molecules. And as an example, at Twist we offer two things. Starting at the bottom is our hard-to-drug target discovery, using our libraries that are inspired by the human repertoire. We've been able to show that we can very effectively pan those libraries and find leads even against hard-to-drug targets. And then at the top, if you already have an antibody, we're able to optimize that antibody against using the power of our libraries. We can design in the computer massive libraries, synthesize them, and then bind them to find either biobetter or finding molecules that have better drug-like properties. And we've done that at Twist for many different targets. We've done GLP-1R. We've done a number of targets in immuno-oncology like PD-1, TIGIT, CD3, ADORA2A, and we also have done it against COVID-19. On the basis of those great data, we've been able to sign a number of fantastic partners in many different modalities, in many different indications. And if we look at the statistics of what we've achieved at Twist, we've completed 19 different programs, and we still have 43 programs that are active with 31 partners. And again, the indications can be very broad, from cancer to neuro to immuno-oncology, infectious disease. We even do drug discovery and optimization for cats and dogs. And then we have very varied modalities, starting from VHS, antibody, bispecific, ADC, TCR, and more. And as I mentioned, but the root to achieve those results is our ability to make amazing libraries. Because we have a silicon chip where we can make millions of oligos at the same time, we can create explicitly millions of genetic elements that have any mutations that you want. And then those elements are super precise. So it's a base by base precision. It's a codon usage precision. We can also precisely do combinatorial assembly of those elements. We can control super precisely the ratio of amino acids. We can control the length of the domains, the length of the CDRs. We can remove restrictions side. We can remove unwanted motifs. We can also pop in that genetic material into many different scaffolds, so that you have combinatorial of CDRs, but also you have combinatorial of scaffold. And then at the end, everything is NGS sequence, so that you know that the quality is there. So again, that is at the root, and that makes sure that you have basically a synthetic library that is human in content. So it's a semi-synthetic library, which is the best of both worlds. And if we contrast those libraries against other libraries like NNS, NSK, or TRIM, we are the only one that can match the natural human repertoire and that can remove the liabilities from the libraries. So now that we've talked about discovery and optimization, we can very briefly talk about everything that we do, from target identification to target validation to drug discovery to drug development and even to disease stratification. We have the tools that Twist, from our genes, oligo pools, libraries, our NGS kits, and our biopharma products. We have the tools to enable you, help you get to your answer. And so we look forward at Twist to working with you and enabling you to get through those stages and write the future together. Thank you very much.
The Cartera LSA platform is a revolutionary and versatile, high-throughput antibody discovery platform that combines patented continuous flow microfluidics with high-throughput surface plasma and resonance to deliver up to 100 times the data in 10% of the time and with only 1% of the sample requirements compared to other systems. The LSA enables you to automate key applications in single unattended runs, including capture-based kinetics assays and epitope binning for interrogating up to 384 by 384 pairwise interactions. The single flow cell sample needle can access a sample block position with various tube formats up to 50 milliliters and a microplate position which can hold a 96 or 384 well plate. The 96-channel printhead can immobilize up to 384 distinct ligands to the array. Vertical bidirectional flow in the flow cell enables extended contact times during immobilization to concentrate material onto the surface of the array. The single flow cell covers the entire array, enabling a simultaneous analysis of up to 384 binding interactions. The LSA can collect fully referenced kinetic data for 384 clones simultaneously. Industry-leading data analysis software enables kinetic characterization and epitope binning analysis in minutes. The Cartera LSA software features an intuitive user interface designed to help you set up your experiments quickly and efficiently. With epitope binning software, data are linked across panels to facilitate review of sensorgrams, heat maps, networks, and a dendrogram. The ability to merge orthogonal data and view networks by color options provides a powerful and intuitive way of assessing your antibody panel's diversity while keeping an epitope-centric view. To learn more about the Cartera LSA and how you can use it to generate 100 times the data in 10% of the time, using only 1% of the sample compared to other systems, visit us online at cartera-bio.com. The Cartera LSA platform is a revolutionary and versatile, high-throughput antibody discovery platform that combines patented continuous flow microfluidics with high-throughput surface plasma and resonance to deliver up to 100 times the data in 10% of the time and with only 1% of the sample requirements compared to other systems. The LSA enables you to automate key applications in single unattended runs, including capture-based kinetics assays and epitope binning for interrogating up to 384 by 384 pairwise interactions. The single flow cell sample needle can access a sample block position with various tube formats up to 50 milliliters and a microplate position which can hold a 96 or 384 well plate. The 96-channel printhead can immobilize up to 384 distinct ligands to the array. Vertical bidirectional flow in the flow cell enables extended contact times during immobilization to concentrate material onto the surface of the array. The single flow cell covers the entire array, enabling a simultaneous analysis of up to 384 binding interactions. The LSA can collect fully referenced kinetic data for 384 clones simultaneously. Industry-leading data analysis software enables kinetic characterization and epitope binning analysis in minutes. The Cartera LSA software features an intuitive user interface designed to help you set up your experiments quickly and efficiently. With epitope binning software, data are linked across panels to facilitate review of sensorgrams, heat maps, networks, and a dendrogram. The ability to merge orthogonal data and view networks by color options provides a powerful and intuitive way of assessing your antibody panel's diversity while keeping an epitope-centric view. To learn more about the Cartera LSA and how you can use it to generate 100 times the data in 10% of the time, using only 1% of the sample compared to other systems, visit us online at cartera-bio.com.
Good morning, ladies and gentlemen. Welcome to the 2024 Therapeutic Antibody and the 19th Frontiers in Biomedical Sciences Conference. This conference is organized by Taiwan Antibody Association, Biomedical Translation Research Center, and the Lin Rongyao Foundation. We would also like to thank all of the sponsors for making this conference possible, and their logos are shown on the posters, banners, and also in the handbook. Okay, now let's welcome Dr. Hanzhong Wu, Chairman of TAA and Director of BioTrack, to give us an opening remark. Please join me to welcome Dr. Wu.
Dear honorable guests, ladies and gentlemen, welcome to Biomedical Translational Research Center, BioTrack, at the National Biotechnology Research Park, NBRP, Academia Sinica. It is my great honor to be here to express my warmest welcome to our esteemed guests to join this antibody therapeutic and the frontier in biomedical science conference, organized by Taiwan Antibody Association, TAA. We are grateful to have you join this wonderful event and share your knowledge in this exciting field. As we gather here to explore the innovative technology and the applications in antibody-based therapeutics and biomedical science, we hope to gain new insight and a perspective on the current trend in the industry. This conference will cover a broad range of topics. It's including antibody drug discovery, antibody engineering, immunotherapy, and the precision medicine. So we believe this event can serve as a platform for all participants to share their research findings and establish a meaningful collaboration. I would like to express my gratitude to all distinguished speakers for their contribution to this conference, as well as our organizer and the sponsor for their kind support in making this event possible. We hope this conference will be a valuable experience for all attendees. I look forward to a productive exchange of knowledge and idea, and to establishment of collaboration between academic and industry. Once again, welcome to BioTrack at NBRP, and thank you for joining this wonderful conference. I wish you a successful and enjoyable experience. Thank you.
Okay, let's thank Dr. Wu. Okay, Dr. Wu, please remain on the stage. Now we would like to invite all the speakers, moderators, and the board members of Taiwan Antibody Association and Lin Rongyao Foundation to come up to the stage for a group photo, please. We will take one here, and then we will. The photographer will come up and take one backwards, so please. Yeah. Please move a little bit so you won't be blocked by this podium. Move. I think you need to move forward a little bit. Okay. Okay, please look to the photographer. Okay, 1, 2, 3, smile. Okay, let's take one with your thumbs up, please. Okay. 1, 2, 3, smile. Okay, thank you. Please take your seats. Okay, now the photographer will go up to the stage and take a photo from the stage. So for the guests sitting in the back, could you please move to the center or to the front? Okay. Okay. Okay, please look at the photographer and smile. Okay, let's have a thumbs up again. Okay, thank you. Okay, now we will start our first session, and let's welcome the moderator for the first session, academician Dr. Andrew Wang Huijun 院士.
Good morning. I'm very pleased and also very honored to be here to moderate the first honorary speaker, plenary speaker, Professor Ang, and An Zhiqiang 教授. Professor Ang, of course, has been sort of interacting with us for many years, actually. So here's the slide. It's not very clear. Maybe you can dim the light. And this was one of the first antibody therapeutic conference in 2015. And you can see Professor Ang is hidden behind the, and you will see many familiar faces here, and including Liu Fuzhong, our former vice president, and Yang Anshui. In fact, he's an expert in phage displayed antibody and theoretical biologist. But I don't know where he is now. I think he's in the United States somewhere. But in any event, there are many, many people here. And I founded this association in 2012. So I'm very glad that the association survived till today. In fact, it's thriving. And so this association actually kind of provide a opportunity and platform for many of the scientists and industrialists to work in the antibody areas. So this conference actually not only serve the opportunity for us to exchange many scientific ideas and so on, but also provide opportunity for some of our scientists to translate our results to applications. And one of the example I remember, I'm not sure whether it's in this 2015 conference or not, but at that time, Seattle Genetics was invited to the conference, and they heard the story from Zheng Tianlu. Where is Zheng Tianlu? There. And about his technology in this lockhead antibodies. And they were very impressed. And subsequently, they interact with Professor Zheng. And now Professor Zheng is one of the richest scientists in Taiwan because they have a very bountiful agreement. Anyway, so today, I'm pleased to have the opportunity to have Professor Ang here. And Professor Ang has been with us many years. You can see that 2015, he already participated in this conference. And I was impressed at that time. He edited a very large book on monoclonal antibodies, and it sort of served as a Bible of antibody therapeutics and application and theories and so on. So he came and exchanged with us, and then at that time I think I was the co-director of NRPB, and we visit Houston and interact with them. But I don't remember much of the detail of Houston. All I remember is I lost my iPhone on the China airplane. So I was disconnected with the communication was outside for several days. But in any event, Professor Ang, we met there, and he also came to Taiwan. And he is currently the director of, as you can see from his CV, of this, I need to be correct, Texas Therapeutic Institute, okay? It's an institute dedicated to drug discoveries and in many aspects. And he's also chair of this Robert Welch Distinguished University Chair in Chemistry at Texas Houston University Consortium. Anyway, it's one of the largest biotech park in the United States. So he obtained many awards, and last night we discussed about the research, and I think we all agree that translation is only an aspect that based on a solid foundation on basic research. And so he has published many papers. He mentioned that maybe the past 10 or 15 years, he has over 40, we call it CNS papers, okay? So it's very impressive. Now he's actually been promoted to the, let me look up your card, otherwise I don't. He's the Vice President of Drug Discovery of UT Health in Houston. So now he's the Vice President in charge of drug discovery, translations, and many important duties at the Houston. So today he's going to give us a talk on some of the developing antibodies targeting, well, I don't know how to pronounce this, LIBRO2 and TRIM2 in Alzheimer's disease. Professor Sang, thank you.
Thank you, Professor Wu, for inviting me for this conference. Also, Professor Wu, very nice to see you again. Actually, my first time here actually was in 2012, the very first antibody drug discovery conference organized by the Taiwan Antibodies Association. So I actually witnessed the establishment of the association some 12 years ago. And so for this audience, it's very clear that antibodies became a major drug modality in the last 30 years. In 2021, we celebrated the 100th antibody drugs approved by the FDA. And so today, just a mere 3 years later, we have more than 200 approvals. But you look beyond the numbers, most of the antibodies are still focusing on oncology and immunology. So today, we shift our effort to also try to address unmet medical areas such as neurodegenerative diseases and diseases in the CNS. Those include Alzheimer's disease, brain tumors such as glioblastoma and glioma. The difficulties of targeting central nerve diseases by antibodies is that antibodies cannot readily cross the brain-blood barrier. So that's limited the delivery of the drug. And another thing is, of course, is, the biology of these diseases are not very well studied. So we do not have very good drug targets for these diseases. So what I'm going to do today, in the next 20 minutes, is to tell you a long-term study in my lab working on target identification and generate antibodies as probes to validate some of these targets for neurodegenerative diseases. Today, I'll show you an example of particularly in the area of Alzheimer's disease. So the first example is called ALRB2, which is an immune checkpoint protein. Show you here, you see this protein has 4 RG-like domains. These are hallmarks of immune checkpoint proteins. Intracellularly, they have 3 IT motifs. These are immune checkpoint inhibitory receptors, such as the PD-1. And so my lab, in collaboration with Alec John's lab at UT Southwestern, have been working for the last 15 years systematically to study this family of receptors. Actually, 2 of these antibodies targeting these receptors got into phase 2 clinical trials for oncology indications. So in 2013, there was a publication in Science showed that the human ALRB2 receptor is a receptor for the A-beta plaque protein, which is the mouse homolog PLB, which regulates synapse plasticity in Alzheimer's animal model. So for the first time, suggested that this family of receptors might play a role in Alzheimer's disease, in addition to cancer. And in 2021, there's a large genetic study, the genome-wide association study showed with over 1 million individuals identified for Alzheimer's patients. They identified 38 risk loci, and ALRB2 is one of the 38 risk factors. And in 2022, 2 years ago, in another independent genetic studies showed ALRB2 indeed is a risk factor by analyzing over 100,000 clinically diagnosed epoxy AD patients. More importantly, in this study showed that ALRB2 is overexpressed in microglia, and is a function is phagocytotic activities of the microglia in the brain. Independently in 2018, so another, the second example I could give to you is called TRIM2, which is a very well validated AD risk factor. So TRIM2 stands for triggering receptor expressed on myeloid cells 2, which you can tell is it has a 1 RG-like domain, which is a hallmark of immune checkpoint proteins. But TRIM2 does not have a functional intracellular domain, and it needs to be partnered with adaptive protein, DAP10 or DAP12. Show you here is DAP12, which provide R-time motif. R-time is a motif that's a hallmark for immune activating receptors, such as CD28. And so R-time adaptive proteins do not have extracellular domains. So 2 truncated receptors, they come together by the positive charged lasting residues with the negative charged aspartic acid residues in DAP12. So these bridge two receptors in proximity, and then constitute one functional immune activating receptor. So what TREM2 does is overexpressed microglia, and it drives microglia's phagocytotic activities to clear debris. So metaphor for this would be, so the microglia is like a garbage truck, so move around in the brain to clear up debris, including A-beta peptides and lipid membrane debris such as phosphatidylcholine and phosphatidylserine. So the TREM2 actually is the gas pedal for the truck. So if you want to move the microglia faster, more active, so you can activate the TREM2. And in 2018, in collaboration with Huaxi Xu's lab at the Berman University, San Diego, so we reported for the first time actually the A-beta peptide is also the ligand for TREM2. And so with the publication size of our publication in 2018, we demonstrated that LRB2 and TREM2, they are all expressed microglia cells, and they share ligands. In this case, they share ligands as the A-beta amyloid protein. And so this was actually validated in this study here. And show you here is where you activate the microglia cells with the A-beta protein, peptide protein. You say the LRB2 can play a role in the phagocytosis. And this blue line is TREM2. And this show you, this is another ligand of LRB2 and TREM2, which is called phosphatidylserine, which is a membrane protein lipid fragment with also debris. Because in the brain, you have a lot of neural damage and apoptotic process. So microglia actually need to clear up the debris, including beta. So with this kind of finding, we hypothesize that maybe LRB2 serves as the immune checkpoint to regulate TREM2. Because we know the fact that, for example, PD-L1 can inhibit PD-1, right? So in this case, our hypothesis was that microglia is the truck, right? The garbage truck. So TREM2 is the gas pedal. So TREM2 actually is the brake for the microglia, because you don't want the microglia to be too active under normal physiological condition. So to test that this hypothesis, this is actually a brain staining of AD patients, and you can tell. So this is a staining of the brain for LRB2, and IBA1 is a microglia marker in purple, and 6E10 is the plaque marker, and TREM2 in red. If you merge it, so you have LRB2, TREM2, they co-localize microglia cells, and the microglia cells actually surrounding the A-beta plaque. And in the bottom is the control, and this GFAP is a marker for astrocyte, which is another cell type in the brain. Show that indeed LRB2 and TREM2 are microglia specific, so they are not expressed astrocytes, in fact not expressed other neural cells. And what we did next was to say if this hypothesis is real. And so we constructed a reporter cell line. So this cell line is two 97 cell line, HEK297 cell line, then you have the TRIM2 RG-like domain, extracellular domain, fused with the DAP12 R-type motif, which is constitute active immune activating receptor. And that will co-express another receptor. This is the ALRB2. So the idea that is if you give the common ligand, say beta peptides, we bind to both receptors, will bring the both receptors to proximity, then ALRB2 should be able to inhibit the TRIM2 function. And in your control construct, what we did was you still have the TRIM2 receptor, but in the ALRB2 construct, we mutated the 3 tyrosine kinase residues. So that basically this muted receptor should abolish the ALRB2 activity. So it's a negative control. The positive control, of course, is the cell that overexpresses only this functional chimeric TRIM2 DAP12 receptor. So sure enough, if you use the oligo beta peptide as a ligand to activate TRIM2, you say the TRIM2 beta activation. This is a GFP reporter cell assay. And if you have the TRIM2 receptor with the mutated, the tyrosine kinase mutated ALRB2, you say no effect. So the TRIM2 can still activate, the ligand can still activate TRIM2. But if you have the wild type ALRB2 receptor together with TRIM2 receptor, with the co-ligand, you say you block the TRIM2 activation of the microglia cell. So you indicate that indeed ALRB2 serves as a negative regulator of TRIM2. This kind of finding is not new. Actually, we know is immune checkpoint immunology, cancer immunology field. Oftentimes, inhibitor receptors function as inhibitors for activating receptors. And this is also true for using the phosphatidyl serine as the ligand. You say the exact same result. So next thing we did was generated engineered blocking antibody that bind to ALRB2, which interferes with the ligand binding. So this is basically ligand receptor blocking antibody. And so if you do this, so this antibody, neutralized antibody, can inhibit the ALRB2 inhibition of TRIM2, as I show you here. So the antibody can effectively rescue the ALRB2 inhibition of TRIM2 activity in both beta peptide or phospholipid serine as ligands. And in this study, we use this human microglia cell line, HMC3. You can tell that indeed both ALRB2 and TRIM2 are overexpressed in microglia cells. And if you treat with this is a beta peptide lipid complex uptake through phagocytosis of the microglia. In this case, we use this human microglia cell line, HMC3. And you see the antibody, this antibody can block about 25% of the TRIM2 mediated phagocytosis of microglia cells. Because of lack of true mouse homolog of ARB2 B2 model. So what we did here is a in vivo study. Is we take a 5-month-old 5X FAD mice. These are transgenic mice, overexpress human A-beta peptides. These mice actually at 5 months old, they not only accumulate A-beta plaque, but also exhibit memory deficits, which is similar to AD phenotype in humans. And so we inject a primary human microglia cell together with the ARB2 antibody through a stereotactic injection. And you see our top panel here, the IBA1 again is a marker for microglia cell, and ARB2 expression at 6E10 is the plaque. And you see control on the top. When you treat with the ARB2 neutralizing antibody, you see a significant activation of the microglia, which is stayed in gray. And this also showed, because the ARB2 is a receptor expressed on microglia, so you see the significant application of the microglia cells. And they indeed co-merged, localized with the A-beta plaque. And in the bottom panel, you show the CD68 is a phagosome marker, which show the activity for phagocytosis. Indeed, this result show you that there's a significant phagocytosis activities when you treat with the ARB2 antibody. So currently, we already generated ARB2 transgenic mice. So we study this antibody in these transgenic animals. Independently, 2 months ago, Marco Canola's lab at Washington University in St. Louis, and he published a paper showed that ARB4, another family member of this receptor family, should have they all expressed microglia, and actually they serve as a risk factor for AD. So this, I think recent trend is emerging that this family of receptors, ARB families, has 5 members, 1, 2, 3, 4, 5. Also, a related member called layer 1, play a significant role in neurodegenerative diseases, including Alzheimer's. But I already told you about the TREM2 as one of the few validated risk factors for Alzheimer's disease. And in this study, this is actually you together with the ARB2 antibody program. What we did was try to generate antibodies targeting TREM2. In fact, there are already antibodies targeting TREM2 in clinical trials. It's basically the agonist antibody. So basically, what I told you, you activate the TREM2, you can increase the macrocytotic activities of microglia to help to clear up the debris in the brain. And the problem with that is the EC50 of these antibodies in in vitro assay is about 100 nanomolar range in vitro. And typically, the antibody without, because the brain-blood barrier, typically you can only deliver antibody to the brain is about 1 nanomolar. You can tell that antibody with 100 nanomolar EC50 in vitro, you can only deliver 1 nanomolar. That's way below the pharmacologically relevant dosing level. So our objective was to engineer antibodies, say we can increase the TREM2 antibody activity in vitro, and also maybe engineer antibodies to help the antibody to get inside the brain. So we did that. So we pioneered the TREM2 extracellular domain of a library, and through phage ladder positive, that the antibody need to bind to the cell surface of TREM2. And we want to engineer our antibodies that do not interfere with the normal ligand binding, because otherwise you're going to have potential side effects. And so we identified one antibody that's bound to the TREM2 receptor on cell surface, the function as an agonist. So I show you here. So these antibodies, so the 11 antibodies, eight of them bind to the cell surface expressed receptor, and four of them actually interfere with ligand binding. Only one that's the agonist. So that's how you screen for antibodies. Antibody AT is the agonist, which show you this functional reporter assay. And indeed, this antibody, if you add this antibody to the microglia, you a beta lipid phagocytosis assay, you see the antibody dose responsive manner can increase the microglia phagocytotic activity. The next thing we did was because to activate TREM2, the TREM2 receptor need to be clustered. So we hypothesized that maybe we can engineer an antibody which we can double the binding site. So normally IgG antibody is bivalent, right? The bind to two receptors, yeah. And so what we did is called tetravalent design. And so you see here, I show you 6 different antibody design with tetravalent binding. And to our surprise that the IgG antibody, the TREM2 agonist number 18, it has the EC50 about 80 nanomolar, which is not different from the one in clinical trials. But if you simply duplicate the binding site, you see that this x-axis, you see the antibody concentration in log phase and the activation of the TREM2 activity. You see all the tetravalent antibodies have a significant improvement of the activity. And the best one is called the TVGD. This has a 0.42 nanomolar activity. So 0.2 nanomolar versus the original IgG, which is 80 nanomolar, is 240 improvement in vitro activity. So in another assay, what we did was, when you antibody activate TREM2, it will activate by binding to coupled with the DAP12 receptor, will activate the spleen tyrosine kinase, S1K. And you see the S1K phosphorylation assay, the IgG is 152 nanomolar, and this tetravalent antibody is 1.4. Again, it's more than 140 improvement in this functional assay. Yet in another assay, as I said, is TREM2 overexpressed microglia can help the microglia to move around in the brain. It's called migration. In the migration assay, again, the IgG is 372 nanomolar. By simply duplicate the binding site, we see the TVDIG is 1.2 nanomolar, improvement of over 300-fold in terms of the in vitro activity. Finally, in the last functional assay in vitro, what we did was using the A-beta oligonucleotide complex that mixed with the microglia cells. You see the phagocytosis of this plaque. Okay. And so you see this is a truly functional assay here. The IgG is 229 nanomolar. The TVDIG is 7 nanomolar, about 70 for the improvement. So this actually is really surprising because just by simple antibody engineering, you can improve the in vitro functional assay over 140 in 5 different assays. So the next thing we did was engineer antibody with the TVDRG, and we also added antibody targeting the transferrin receptor, which can help with antibody cross the blood-brain barrier. And you see this became a homologous molecule, antibody engineered molecule. And to show you here, it is by adding this transferrin receptor, monomeric receptor antibody, did not impact the engagement of the TREM2 target, which is mediated by the variable region. So this is the PK assay. A single dose in a mouse, this is RP systemic injection. You see with the transfer receptor, you see significant uptake of the antibody brain, and the antibody with the control antibody here, you see the minimal penetration of the antibody brain, which I told you earlier, it's about 1 nanomolar. And show you here is the CD31. This is the mice injected with the antibody. CD31 is a endothelial marker, marks the vasculature of the brain, and this antibody ABAT targeted antibody, and RBA1 again is the microglia marker. You see this construct with BBB delivery transfer receptor antibody, you see significant penetration of the antibody in the brain, and better yet is actually co-localized with microglia cells. So next we did was this functional in vivo assay, and so we this is again the 5X FAD mice that overexpress human plaque protein, also exhibit human Alzheimer's disease like phenotype. And so we give this antibody actually weekly for 15 weeks as a long-term treatment. And so surprisingly, this antibody is very safe. It's a 20 milligram per kilogram dosing, RP dosing weekly for 50 weeks. We do not see any impact on the body weight or feeding pattern, suggests this antibody would be safe, at least in this particular animal model. So this is the punchline of this study. That so here you see the cortex, also you hippocampus of the mice, and if you treat the mice with the antibody, the TREM2 agonist tetravalent antibody, you see without the transfer receptor antibody, because antibody cannot get in, you see the accumulation of the plaque, no difference. And another control that is antibody with the transfer receptor, but without the TREM2 agonist antibody, you see the significant accumulation of the plaque. And in the bottom, you see that if you treat with the TREM2 agonist tetravalent antibody coupled with the transfer receptor antibody, you see a significant decrease of the beta plaque. And this is just a quantitation of the data. And finally, we want to see if this antibody can improve the cognitive function of the mice. So we did multiple functional behavior assays. I'll show you this here one. In this particular model is called the elevated plus maze test. So normal mice, they are afraid of height. So if you put the mice in the cage, so they will prefer to stay inside this closed the cages here. But there's in the middle of this open cage, there's this bridge, and the 5X AD mice, they kind of not afraid of height. That's a behavioral deficit. And so they actually better this bridge more often. So I show you here that is if you treat with this TREM2 tetravalent agonist antibody coupled with transferrin antibody cross BBB, you see the significant reduction of the mice stays in the open arm. And of course, secondary increase is a close arm for these animals. And to summarize, I'll rush very quickly to tell you two stories in our labs working on validation of AD targets. And I showed you that both TREM2 and LRB2 are potential targets for Alzheimer's disease. And we also discovered LRB2 actually is a brake or negative regulator for TREM2. And you can imagine we can engineer biospecifics for this kind of design. I think the take-home message is, so last night we had dinner with Professor Zhang while we discussing about translational research. And to me, translational research actually demand even more stringent basic science. We need to spend time validating the target. So I myself, as an antibody engineer, actually I would say that anybody can engineer antibody, but not everybody can identify the right target to work on. And so to show you this, so most of the antibodies came from our lab, lasted for more than a decade before the antibody has any potential for drug development. This was published, most of the work is published. The TREM2, the identification of TREM2 is in neuron in 2018, and this TREM2 tetravalent TREM2 antibody published last year in Science Translational Medicine. And with that, I stop here. Have time for any questions?
Sure, yeah. Thank you, Professor Yang, for the wonderful story. As we all know, that Alzheimer's disease is a very hard disease to treat. And so hopefully those antibodies could have some use in the future. I think we can open for a few questions before the break. Anybody? Oh, over there?
Yes.
Dr. Lai?
Yeah, Dr. N, very good talk. Impressed by your research. So you mentioned that LBR2 actually able to regulate TREM2 for the activity. So later on, you developed bispecific antibody with TREM2 with the TFR, and looks very efficacious. I'm just wondering that. What do you think about developing bispecific antibody against LBR2 and TREM2? That on one end activate, in the other end block the LBR2 activity.
Yeah, no, that's a possibility, right? So, so you can imagine. So the bispecific they will become a tri-specific, but you need to have the transfer receptor. The TREM2 antibody need to have a tetravalency because. Then you have the ARB2 antibody. So the molecule could become too complex for drug discovery purposes. I think that's something I think academically we need to explore. And you never know, maybe as a drug is even though it's complex, but we can still. We need more work on that. That's a very good question. Yes.
Professor An, thank you for sharing the very exciting data. This is Ruby Chen from Genomic Research Center, Academia Sinica. So I have two questions. The first is, what do you think about the treatment window of these kind of antibodies to patients? Because we know that if maybe too much phagocytosis may lead the microglia become some, disease, more activated microglias, and may be bad for the patients. Yeah, and the second question is maybe relatively small, but when you do the phagocytosis assays, you always use the A-beta oligomers with the lipid complex, but usually in the plaque, it's just A-beta. It's not that much of lipids. So I just wonder, why the assay need a lipid?
Yes. Yeah, those are good questions. So the first question first. So Alzheimer's disease, as you know, there are two antibodies approved for treating of Alzheimer's disease by the FDA. The first one is Biogen, and the approval actually was very controversial because Biogen already presented data for plaque reduction, but did not present data for cognitive improvement. But FDA nevertheless approved the drug. We can discuss that afterwards. It's very controversial. But at the same time, actually, Biogen gained over decades of experience in clinical studies of Alzheimer's disease. So they actually team up with Eisai, Japanese pharmaceutical company. The antibody to me is almost the same. It's just a butt plaque, remove plaque. So in this case, they selected early onset AD patients in the clinical trial. Sure enough, there's not only plaque reduction, but also cognitive improvement. So this comes to your question, that is, for any AD drug development, we want to target early onset disease. In fact, the treatment tool is actually detrimental in late onset disease, as you know. So actually, we also generating antibodies, which is an antagonist treatment tool for late onset development, because that time overactivation of microglia will also damage normal cells. And so your question about A beta oligo-lipid complex, because in the brain, the plaque is an oligomer of A beta protein, not lipid complex. But for in vitro assay, to oligomerize the A beta peptide, so we use lipid to mimic the in vivo situation. So it's a in vitro model for what's happening in vivo.
I have a question regarding the treatment of the Alzheimer. Now, if you treat the patients and let's say the plaque reduce and cognitive ability recover somewhat, but if you stop the drug, would it come back? Mm-hmm. In other words, do you have to treat the patient whole lifetime? Yeah. And that will be very expensive, yeah, especially for antibodies.
Yeah. So before I answer your question, so let's revisit, for example, like a PD-1, right? So the idea that is immune checkpoint targeting therapies should have a disease-modifying effect. In other words, has a memory. And but in the clinic, where people treated with PD-1 antibody get a full recovery, the cancer-free, but the physician is still debating, should we stop the drug altogether or we should continue? And the different, you get papers every day discussing that. In terms of Alzheimer's disease, so for the A beta targeting antibody, those you need to give them all the time because it's really do not treat the mechanism of disease. A beta is many people consider is the result of AD, not the cause of AD. But if you target the ARB2 treatment tool, which is immune checkpoint targets, and potentially, actually, in our Science Translational Medicine paper, we also show that activation of a treatment tool can increase the neural survival, suggesting that these antibodies may have a disease-modifying effect. Therefore, you can treat it once, may last for a long time. But those are just hypotheses. We need to test it.
Do you have an estimate how much cost millions of dollars for the whole life treatment?
Yeah, actually, for the A beta antibody, it's very expensive. It's about $150K per dose of treatment in the US. And so there's many clinical trials. Another thing I will talk about is BBB. So the antibodies targeting the beta peptides actually is normal IgG1. There are no BBB penetrating capabilities. So what people do actually is you give the antibody through systemic injection. and use the ultrasound to actually radiate your brain, you can temporarily open up the BBB.
Yeah.
Actually, this is very successful now. So let's develop. I think the question is, you can develop drugs and worry about the cost later.
Okay, any further questions for Professor Yang? Okay, if not, let's thank Professor Yang for his wonderful talk and to end the break.
Okay, let's thank Professor An and also the moderator, Dr. Wang. Now let's take a short break because we're a little bit behind the schedule. So let's come back at 10:40. And we have prepared coffee on the third floor, so you can go to the third floor to enjoy coffee and also visit the sponsor booth. Thank you. We will resume the conference soon, so please take your seats. Okay, we will now resume the conference. And for the next session, let's welcome our moderator, Dr. Zhang Ziwen.
Okay, I'll just stay down here. We'll move on to the next two presentations. I'm Chae-Wen Chen, from the company ImmuneWork and TE Meds, located in this building. Well, for the next two presentations, I will moderate the presentations and also introduce the speakers. For the first one, Dr. Jen He, He Zhenhong, okay, a company also located in Taipei. Dr. He's experience and his past achievements are very well described in the brochure, so people can refer to the description. But I want to say something about the company. Dr. He founded AP Biosciences. I think today he will talk about the technology and other aspects of the company, AP Biosciences. I know that company well, and I will say this company, AP Biosciences, is exemplary among the many biotech companies in Taiwan. The company has a very novel technology. They have a few products in the pipeline, and the company can move these products rather expeditiously into preclinical development and into clinical trials. They also have a very good business development strategy. They are working well with other companies, for example, with API. So in various aspects, this company is doing very well. And I think their technology, especially the bispecific antibody technology, are rather innovative. Today we'll hear about that part. I think I just talk about AP Biosciences and Dr. Jen He now, and I leave the introduction to the next speaker later. Okay, Dr. He, please step up here. 在我后面。Okay, please.
All right, thank you, Dr. Zhang, for the very kind introduction. Since the major part of our audience speaks Chinese, so I'll do my presentation in Chinese as much as possible. All right, 我稍微解释一下整个，因为原想我们是在开发所谓的抗体药物，尤其是 focus 在所谓的双特异性抗体。那前面几张 slide 我就稍微简单介绍一下整个抗体的市场，它的整个 big picture。从1986年第一个therapeutic antibody OKT3 被 FDA approved 到现在，其实已经有超过100多个抗体拿到药证在市场上。那主要的适应症当然还是以癌症跟免疫，包含 autoimmune inflammation 这些为主。那如果说你从这些上市的抗体药物的市值，它的营业额来看，几乎每年过去10年前10大的 best selling drugs 至少5个到6个都是抗体药物。 包含去年卖的最好的 Keytruda PD-1 的 antibody for different cancers， right， 在去年卖了$250亿。第二的 Humira 是 for autoimmune， 它其实在去年还是 best-selling drug， 今年它的营业额降到140多亿，主要是因为一些 biosimilar 今年已经开始出来的 competition。所以你可以看出来，前10大2个就是抗体药，其他有3个是免疫。那比较特殊的一个是艾利亚，它是一个眼科用药，它基本上是针对所谓的湿性黄斑部病变呀。那这个市场因为 aging population 越来越大，所以这个市场也越来越多。目前有3个所谓的 anti-VEGF 的 antibody drugs on the market。去年总共的营业额大概$130亿。所以 just give you a quick idea how big the market is。好，那这个是 projection of antibody sales。你可以看到它的快速的成长。Okay? 那我们说Keytruda去年250亿，我这里只是稍微做一个 comparison。台湾的护国神山， okay, 去年的营业额是$690亿。Okay? 所以如果说台湾能够开发一个类似Keytruda这样的药物，那我想可以称为护国神药。Okay? 那因为抗体市场非常非常的 robust， 而且 expanding。所以事实上，前10大的所谓的 pharma companies 全部都 involved 在 antibody development。好，那如果说从1986年第一个 OKD3 在上市之后，到现在差不多40年抗体的历史，你以每一个 decade 每10年来看， okay, 第一个10年其实被 approved 的 antibody 不多，因为 technology 刚刚开始 mature。可是第2个10年，它加了变成8倍，第3个10年又增加了，真正的 jump up 是在过去10年。Okay? 那我相信这个 trend 会继续增长。未来10年可预期的，因为这些基本上大部分来自所谓的 first generation antibody drugs， 也就是说所谓的单特异性抗体。那未来10年我们会很期待看到什么 second generation antibody drugs， 那包含我待会要讲的 ADC、CAR-T 跟 bispecific antibodies， 还有所谓的那个 degrader antibody conjugate。那它的市场我们刚刚说了，前10大主要是在 cancer oncology 跟 autoimmune inflammation， 所以你也可以看得出来，目前大部分被 approved 的药也是集中在这2个领域，因为目前这个市场最大。那其他的事实上 poses as a unmet medical need， 所以很多公司反而譬如说反而会去针对这些 target 去 develop 他们的抗体。好。那我刚刚提到130多个 FDA-approved antibody drugs， 大部分都是所谓 monospecific antibodies。They work, right? Keytruda $25 billion， 可是事实上还是有大概80%的病人没有办法 benefit from Keytruda。Okay? 所以大家其实在很早就开始思考，can we make antibody better? What can we do? Okay? 所以开始大家的思考就是，在 monospecific antibody 上面再加 second function， okay? 所以有所谓的 antibody plus one 是这个意思。Okay? 那这里面大概有可以归类到这几类了，大概bispecific antibody drug conjugate、chimeric antigen receptor T cells， 这个是比较新的东西。Okay? 我们一般来说 ADC 指的是 monospecific antibody 加一个 chemo drug， 可是现在开始大家在开发的是bispecific 上面再加一个 chemo drug， 就 bispecific ADC。去年有一个 licensing deal， 大概是临床前 phase 1的一个 bispecific ADC， 整个授权金的总值超过$80亿。那另外 antibody cytokine fusion 这个其实也是一个蛮热门的地方，还有degrader antibody conjugate。那我今天会 focus 在 bispecific antibody， 那下午浩鼎的赖研发长会跟大家介绍他们的 ADC 的 program， 然后那个安宏的陈研发长会跟大家讲 他们的 DAC program。好，那所以 bispecific antibody，I mean， 坦白说，从学理上来探讨，你至少有大概70-80种不同的方式可以去create 一个 bispecific antibody。可是事实上，真正进入临床被 approved， 大概在10个左右。那原因很多 ，OK， 那在这里我们稍微看了一下双抗的基本结构，在这70-80种里面，你大概可以用这4个来做分类。一个就是它的结构到底是左右对称呢，还是不对称？ okay? 另外一个是它的 antigen binding 到底是 monovalent 还是 bivalent， 就单价或者双价的结构。第三个，他们对2个 antigen binding 是同向，比如说在这里是面对同一方，还是像这样子的结构是反向的 ？OK， 那第4个，有些 bispecific antibodies 有 FC region，OK， 有些没有 ，OK， 那为什么会做这样的选择？他们的考量点在哪里？基本上，第一个，你要了解你的 bispecific 的 target 到底是什么？是 soluble ligands 还是说membrane protein？ 那 membrane protein 是 on the same cell 还是 on different cells？ 这些都会影响到你的选择。另外一个很重要的就是 ，What do you want your bispecific antibody to do？ 它的作用机制是什么？你到底希望它能够达到什么样子的效果 ？OK， 这个会决定你应该用什么样的 structure 去 create 你的 bispecific antibody。当然 ，efficacy, safety is always a concern。那原想我们主要的是 focus 在这样的一个 structure。那待会我会做一些更详细的介绍。好，那我们刚刚讲有各种方式去 create bispecific antibodies。那这个市场到底有多大？到底有几个 antibody 被 approved？ 到目前为止12个。OK， 那前面2021年之前其实只有3个，也就是说，相对于 monospecific antibody 1980年到现在130多个，真正 bispecific antibody 进入市场上大概在10年前，第一个是最早进入市场是 Amgen 的 Blincyto。Okay， 它是一个 anti-CD19、anti-CD3 的 T-cell engager。接下来2022年，你突然间看到 approved 了4个 bispecific， 去年也有4个，今年到目前为止有1个，也就是说这个 trend 慢慢的这个 bispecific antibody 慢慢被市场接受，那也经过 regulatory 的一些 challenge， 所以它整个 bispecific 的市场的价值，从去年的大概8点多9个 billion， 预测到2030年的时候会超过110个 billion。Okay， 好，这是整个市场，那表示 antibody 在市场上有在做贩售。那可是有些公司，像一些 biotech company， 它 develop bispecific antibodies， 它没有办法一路把它带到市场上，所以它在某个 development 的 stage， 它会 prefer 做 out licensing。那到底这样子的 bispecific antibody 有没有价值？在过去4年，2020年到去年12月，大概有12个 major 的 bispecific antibody licensing deals， 那其中有9个 ，I'm sorry， 有14个。其中有9个是在 pre-IND stage， 2个在 phase 1， 3个在 phase 2。你可以看到他们的 upfront payment， 还有 overall 的 deal value 其实都 very lucrative。所以 that give you some incentive to develop bispecific antibodies, right？ 好，那这些目前被 approved 这些 bispecific antibody， 他们的 mechanism of action 到底是什么？坦白说，14个里面。12个里面有9个是 CD3-based T-cell engagers。OK， 那它的 mechanism 主要就是一边bind到 T 细胞上面的 CD3， 然后一边bind到 tumor cell 上面的 specific antigen。OK， 利用这种方式把2个细胞带在一起 ，activates T cell， 然后去 kill cancer cells。Ideal, right？ 我们待会会提到底有什么问题。另外3个比较特别 ，Brabant， 它是 target 到 non-small cell lung cancer 上面的2个不 不同的 target， 一个是 c-Met， 一个是 EGF receptor。OK， 然后 by binding 它 block both receptor 的 activation。OK， 它用这种方式去治疗。Vabysmo on the other hand， 它是 bind 到两个 soluble ligands， 一边是 VEGF， 另外一边是 angiopoietin 2。那这两个 factors 都 involve 到new blood vessel 的 formation， 所以它的 application 主要是在湿性黄斑部病变。OK， 那这个从2020年被 approved 之后，去年的营业额就直接跳到2.5、2.6个 billion，so it's taking up the market share pretty fast。另外一个是卖的最好的 bispecific antibody 和 Libra， 它的 mechanism 又不太一样。它的确也是 bind 到两个 soluble factor， 那是 factor 9A and factor 10。那在正常的人里面，它是靠 factor 8去结合 factor 9A 跟 factor 10， 然后造成凝血功能。OK， 可是有些病人身上他可能没有表达 factor 8， 或者说他有 autoimmune 的 activity 去 block factor 8的 function， 所以 Roche 他们 develop 这样一个 bispecific antibody， 它同时把这两个 bind 到结合在一起，用这种方式去 activate 那个 factor 9A 跟 10， 所以它主要的 indication 是在血友病上面。那去年卖了超过40亿美金。好，那这个是刚刚excuse me。我们刚刚提到说12个里面9个是 CD3 based， 你可以看到这8个 CD3 based 基本上都是 for blood cancers 血液肿瘤，唯一一个 for solid tumor 的是这个礼拜刚刚被 FDA approved 的一个 CD3， 它的cancer target cancer cell target 是 DLL3。那它主要的 indication 目前被 approved 是 extensive stage small cell lung cancer。OK， 这是唯一的一个 CD3 based T-cell engager for solid tumor。OK， 其他的为什么不能做 solid tumor？ 你如果看它的 dose， 相对于一般抗体的 dose， 它们这些其实都非常低。好 ，the question is why？OK， 因为抗体一碰到 CD3 的时候，它很快就会把它活化起来 ，T cell 就开始 secrete 一些 cytokine， 所以很容易形成cytokine release syndrome。OK， 所以它的 therapeutic window 没有很大，那它只能目前比较能够治疗的就是血液肿瘤。你如果要做 solid tumor， 基本上你可能要提高你的剂量，这时候它的therapeutic window， 它的 safety profile 可能就没有那么好。Okay， 这是很大的一个问题，所以原想我们看到这个，那我们了解到这是一个 opportunity， 所以我们就选了另外一个 T cell 上面的 receptor 去 create 我们的 bispecific platform。好，在我介绍我们的 platform 之前，大概说一下，你要 create 这个 bispecific antibody， 你要考量的是什么？很多人很直觉的想说，那你就是把两个 monospecific antibody 用 molecular biology 把它接在一起，就是一个 bispecific antibodies。Yes and no，OK， 重点就是what you want to have is not just 1 + 1 = 2， 你甚至不是要1 + 1大于2，你要的是两个 monospecific antibody in combination 没有办法做到的效果。这样子的 bispecific antibody 才有它的价值。OK， 所以在这个情况之下，你就要很清楚的去考虑，你要选择什么样子的 target， 你怎么去筛选你的两个 monospecific antibody， 当它合在一起，不管你最后选择用什么样的 structure， 它可以达到你要的效果。OK， 这个事情要想清楚。OK， 还有它的 valency。跟它的 orientation 会根据你你的 indication， 还有你的 antigen 的选择，会有不一样的选项。Okay， 另外一个很重要的，如果你的目的是想要 cure tumor solid tumor，OK，像我们刚刚提到的 CD33 based T-cell engager， 为什么没有办法做 solid tumor？ 因为它的toxicity 太强，因为它一碰到 CD3 马上就活化了 T 细胞。好，那你有没有办法做到所谓的 conditional activation of your T cells or even NK cell whatever？OK，所以这些是主要的考量。好，那当然这是opportunities。那可是做 bispecific antibody 有一个最大的 challenge， 刚刚安教授其实有提到，你的抗体越complexity 越大，你的 engineering 需要的做的功夫就越强，到最后你的抗体可能在实验室上 work 得非常好，可是当你最后要进入 CMC、进入临床、进入 commercialization 的时候，会碰到很大的问题。那这些基本上我们就把它归类在所谓的可开发性或者 developability。那简单的来说，你的 productivity， 你做出来的 integrity， 什么是 integrity？ 你一个 bispecific antibody 通常不会只有一个 single polypeptide， 你可能有2个，甚至有3个或者4条 polypeptide， 它同时表达在细胞里面的时候，它们如何 assemble 在一起？你能不能达到所谓的 100% assembly？ 你后面的 downstream process development， 你能不能很有效率的把它 purify 出来 ？OK， 这些都是 challenge。最后你做出来这个 bispecific antibody 到底够不够 stable， 包含你的 shelf life， 包含你的 serum stability， 这些事先都要做测试。当然 solubility 有时候是一个考量。举个例子来讲，艾利亚我们刚刚提到，去年卖了130亿美金。它是要打到眼睛里面去抑制微血管增生，它能打进去的量很少，大概50到100个 microliters， 可是你希望给很高的剂量，这时候你的 formulation 就很重要，你必须要把这个 antibody 的 solubility 提高到比如说200个 milligrams per ml， 你才有办法达到这样子的剂量的要求。OK， 所以 solubility 很重要。那同时，当你 concentrate 一个 protein 的时候，有些 protein 它很容易进入 aggregate， 那这个会产生一些不必要的 side effect， 所以这些都是你事先必须要考量的事情。好 ，so how does an antibody work before we get into bispecific？ 这大概是主要的 antibody 的functionality 跟 activity， 一个就是 target 到你的，比如说 membrane bound receptor， 另外一个是 blocking， 它可能 block 2个 free ligand， 或是 block 你的 antibody 一个 ligand， 或是 block 你的 receptor。那 binding upon on the receptor could be antagonistic or agonistic, depends on the binding site and the property of your target。另外一个，如果说从cancer therapy 的角度来看，你会希望 upon binding to the target cancer cells， 你可以 introduce 你的 ADCC 或者 CDC 甚至 ADCP activity to kill the cancer cells。所以monospecific antibody 的 mechanism of action 非常的直觉，非常 straightforward。On the other hand, for bispecific, it's very different。好，那所以我们刚刚提到说 Keytruda the best-selling anti-cancer drug in the whole world。25 billion， 可是一样有80%的病人没有办法 benefit from the drug。 Why is that？ 为什么肿瘤很难治疗？这是一个很简单的所谓的 cancer immunity cycle。一开始，你的cancer 上面的 antigen 在 solid tumor 里面，它可能自己apoptosis 或者被 dendritic cell 或者被 macrophage 把它 take up， 然后把这些cancer specific antigen present到 lymph node 给这些 naive 的 T cell。T cell gets activated, expanded, and gets into bloodstream, right? And they start looking for where the tumor is。所以走，好不容易发现 tumor 在这里了。这时候可能发生几件事情。OK， 可能是因为这个 vasculature of the tumor， 或是因为一些cancer 的 stroma、tumor 的 stroma， 你的 T cell 进不去。OK， 好。 那如果说那这个我们把它所谓叫做 cold tumor，OK，好，那好不容易你的 cancer 进去了 ，infiltrate 到你的 tumor 里面，你不见得看得到你的 cancer cells， 因为在 tumor 里面除了 cancer cells， 还有一大堆 stromal cells， 一大堆 immune cells，right？ 那你的 cancer cell 它可能会把它原来的 antigen 把它 block 住，或者把它 shed 掉，所以你可能看不到。所以，那即使你看到了，在那个 tumor 里面也有一些 immune suppressive 的一些 activity 去抑制你 infiltrated T cell 的 activity， 所以看不到，打不到，打不死，这是为什么 tumor 很难治疗的原因。那 bispecific antibody， 像我们在 develop bispecific antibody， 就是要解决这个问题。所以 T-cube bispecific antibody platform 基本上长得就像这样子。我们前端是一个 regular IgG， 后面接一个 single chain antibody， 这边bind到 activated T cell 上面的 CD137， 另外一边bind到 cancer 上面的 specific antigen。OK， 那基本上我们的 platform 这一块已经 optimize 好了，你只要换不同的 targeting 的 Fab， 我们就可以 create 一个新的 antibody bispecific antibody。所以根据这个 platform， 我们目前已经有3个 bispecific antibody 在 develop pipeline。好，那这样子的一个 structure， 这样子的一个 orientation， 还有这样子的 valency， 到底我们希望它做什么？ Okay， 我们希望第一个，我们希望透过这个 bispecific antibody， 把 T 细胞跟 cancer cell 做一个结合，这是第一件事情。所以 by definition it's a T cell engager。好，第二件我们要它做的事情是，只有当这2个结合的事件同时发生的时候 ，T 细胞才会被活化起来。OK， 记不记得 CD3 based bispecific T cell engager？ 它只要看到 CD3， T 细胞就被活化。我们不希望我们的 bispecific 看到 CD37 就活化它。OK， 我们希望等到另外一边 bind到 cancer cell， 所以是一个 conditional activation 的一个概念。所以当它活化之后，它就开始 secrete， 比如说 IL-2 interferon gamma 去induce T cell proliferation expansion， 同时去 secrete perforin 跟 enzyme， 然后去毒杀这个癌细胞。这个就是我们 bispecific platform 主要的目的。好，所以它兼顾了所谓的 safety 跟 efficacy。那另外一个我们刚刚提到的 how about developability？ 像我刚刚说的，其实我们在做这个 optimization 的时候，已经把 developability 的 consideration 全部都放进去。所以像我们现在的 productivity， 我们可以达到4个 grams per liter in GMP setting。那 considering it's almost 200 kilodalton protein, that's pretty good. 那 overall recovery 可以达到 60%。那这个是我们是委托给泰康帮我们做的。CJ 在吗？哦，那个 LC 在这里。Yeah, they have done a very good job for our bispecific antibodies. 好，那它的 stability 也很好。我们事实上放在 PBS 在4度C， 其实到目前为止至少3年的 stability 了。Okay， 好，那我们为什么做这样子反向的一个 binding 的一个结构？重要的就是，你如果说是这里 bind 到 T cell， 这边 bind 到 cancer cell， 事实上你这个可以打开的角度有限制，所以有时候不见得可以很有效率的把2种不同的 cell type 结合在一起。那我们用这样子的方式来做的时候， it has no problem。 同时 it's easier for T cells to create an immunologic synapse. Okay? That's why we choose 这个 structure。 好，所以目前为止这是我们原想目前的 pipeline。 那这2个是已经授权出去的，这个是 for 世界黄斑部病变 HMD， 目前在3期临床。那203、402、601就是根据我们刚刚描述的那个 bispecific T-cube platform create 出来的 bispecific antibodies。AP203 比较 advanced， 目前在1期。 好像还剩10分钟是。AP40目前预计9月要在 Australia file IND， 那我待会就简单介绍一下这2个 program。所以 AP403它的 target 是 PD-1， 那另外一边就是 bind 到 CD137。那它针对的是 PD-1 treatment failed 的 patient。因为我们相信我们的 bispecific 可以比 monospecific work 更好。那它主要的 mechanism， 第一个，即使没有 bind 到 CD137，by binding to PD-1 的 alone， 它可以 block PD-1 的那个 inhibition 或 activation whatever you call it。另外一个是当它同时结合的时候，它可以很有效率的 activate T cells。那不只是，所以在那个 tumor microenvironment 里面，即使有2种 T cell type 非常重要，而且它的量也是蛮多的，一个是 memory T cell， 一个是 exhausted T cells。这2种细胞存在在 tumor cells 里面，那它们为什么没有办法很有效率的 kill cancer cells？ 主要就是它们被这个 cancer cell downregulated in some way。所以透过这样一个 bispecific， 我们希望可以重新再活化这些 exhausted T cells 跟 memory T cell， 让它的 anti-cancer 的 activity 可以last longer。那这里只是秀给你看说，以这样的一个 structure， 我们可以同时 bind 到2个 target。那 this is done by in vitro by Forte Bio。好，另外一个我们刚刚一直提到说，我们是一个 conditional activation target dependent activation。那在这里我们用 HEK cells。with or without PD-1 expression， 去看看我们的 bispecific antibody AP203是不是真的像我们刚刚说的，可以是一个 target PD-1 dependent activation。所以在这里，我们看的是2个 T cell activation 的 readout IL-2 and interferon gamma。你可以看得出来，只有在表达 PD-1 的情况之下，我们的 bispecific AP203才会很有效率的 induce T cell activation。好，那我们刚刚另外又提到 safety，right？ We don't want our bispecific behave like anti-CD3 based T cell engager。所以在这里，我们把 freshly isolated human PBMC 跟我们的 antibody 放在一起，那里面会有 T cells， 可是 presumably 没有太多 PD-1 的 expression。我们看在这个情况之下，当我们的AP203只 bind 到 T 细胞上面 CD137的时候，它会不会 induce inflammatory cytokine 的 expression？ As you can see here, it behaves pretty much like a negative control. As a positive control, OKT3, which binds to and activates CD3, it induces a lot of cytokine expression. 所以好，那我们刚才看到的是一些 artificial 的 cells，right？ HEK transfected with PD-1， 在这里我们用的是pancreatic cancer cells， 然后同时 repeat 刚刚那个activation experiment， 发现的确就我们的 AP203 not either mono or combination could activate T cells。好，那 cell-based function assay how about tumor？ 那在这里我们做了一个那个 lung cancer 的 tumor model， 你可以看到我们在很低剂量的时候，事实上就可以 suppress tumor growth much better than monocombo。好，这个基本上大概就简单介绍一下我们 AP203的 program。那目前是在台湾执行1期临床，有中国医药大学还有台大医院来负责。那下半年我们会加入林口长庚。还有台北医学大学，还有成大。AP402 is very exciting。它针对的 target 很好玩。我们都知道breast cancer overexpress HER2，right？ 然后我们知道有很多 antibodies， 包含 Herceptin、Kadcyla、Perjeta in HER2， either as a monospecific antibody or as ATC could be used to treat breast cancers in HER2 positive。cases。 好 ，so pretty good, right？ 可是不管怎么说，经过这些 antibody treat 之后的病人，还是会有相当大的 percentage 可能30%到50%会有 refractory 的现象，甚至会有 resistance。那我们如何去处理这些问题？那这些问题到底是怎么产生的？那目前临床上大家有看到大概有相当大比例的这些譬如说 Herceptin treatment refractory 的这些 patient， 他们事实上在他们的 HER2 expressing cancer cell 上面发生了一件事情，这个是 full length 的 HER2， 所以 Herceptin could deal with it。可是，在这些 refractory 的病人身上 ，the majority chunk of HER2 extracellular domain is cleaved away， 所以这些 antibody no longer bind to the cleaved HER2 or we call it HER2 variant。Okay, so what are you going to do about it？ 所以我们在原想，我们 create 一个 antibody， 一边bind到剩下来的这短短的一段。另外一边接到 T cell 上面的 CD3 期，然后 create 这样一个 bispecific antibody to try to manage 这个 issue。那这个 antibody 这个 antigen 其实很好玩，剩下来的这一段在 normal cell 上面没有表达。虽然说在很多 normal cell 上面，它的确是有表达 full length 的 HER2， 可是这个 epitope在这些细胞上面其实并没有被 expose 出来，所以 by definition it could be a 所谓的 neoantigen。所以像刚刚 AP203， 我们叫 target PD-L1，PD-L1 在很多正常的免疫细胞上面都有，所以我们的 FC 上面的 ADCC activity 我们是把它 knock out 掉的，因为我们不想要有太多的 safety 的 issue。But in this case, in the AP402， 因为这是一个 neoantigen。So in addition to engaging T cells, we also have ADCC activity and ADCP to help kill the cancer cells more efficiently. 那这个是一个 IHC 的一个 staining， 证明说在很多 patient breast cancer 还有 gastric cancer 的 patient 身上他们的确都有表达这个HER2 variant。好，同样的 structure。as the AP203， 在这里我们可以看到，只有 AP203 not the mono or mono combination can activate T cells。好，那这个 HER2 variant 在 patient 上面做 IHC staining 可以看到，可是问题是，你很难找到一个 stable cell line expressing HER2 variant， 因为它是非常的 kinase。它是一个constitutive active 的一个kinase， 所以很难做一个 stable cell line。可是我们 managed to create 3个 stable cell line in the MD293 cells，231 cells。那同时我们 isolate 3个细胞株，表达高、中、低量，然后用一个 report system 去 check。看我们的 T cell activation activity 是不是 depend on 这个 HER2 variant expression level, and turns out that indeed it has HER2 variant expression dependent the T cell activation activity, which is very interesting. 好，那我们刚刚也提到 safety, right? 像 AP203 一样，我们同时去 check 看我们的 AP402 会不会activate T cells upon binding to CD137. Turns out it doesn't, as expected. Okay. So actually, 我们已经做了GLP 的 monkey 跟transgenic mouse 的一些 tox 的 study. Safety profile looks very, very good. 好，我们刚刚提到4个作用机制。Okay, 那这里只是 show data. Okay, 这里是T cell engagement. 我们看到 AP402 indeed 可以 induce T cell 的 cytotoxicity。这里是 ADCC、ADCP。另外一个我刚刚没有讲的 ，upon binding to HER2 variant， 它事实上也可以 induce receptor 的 internalization。那从这里我们当然很多人就会说，你能不能 create 一个 ADC against 这个 target？Indeed， 我们已经在做这件事情。那已经有一些 proof of concept。它事实上induce internalization 的效果非常好。How about tumor growth inhibition？ 那我们 create 两个 cell line， 一个是在231 cells， 一个是在292。OK， 你可以看到，一般的 monospecific antibody rely on ATC activity， 通常需要到10个 milligrams per kg 才会看到比较有效的 tumor growth inhibition。OK， 在这里我们0.4， 我们就可以看到 TGI above 100%。时间到了哈。我想今天大概就 present 到这里了，那看看大家有没有什么问题。
Dr. Hurst, thank you for your very interesting presentation. Dr. Hurst had an overview of the market and development of the bispecific antibody field. And then he talked about the limitations with the bispecific antibodies with the T3 engagers. And he then went through the rationale how his group developed bispecific antibodies that also engage T cells, but different from those with anti-CD3. And then finally, he talked about the specific products they have and the properties of them and the clinical development of all the biologic activities and the clinical development. So we'll have just a few minutes for questions. 那个
何博士，谢谢你的分享哦，非常厉害的 bispecific antibody。我只是 curious， 这个作用就是说把两个，比如说不同的recognize 不同的 receptor 拉在一起。那 what if the antibody get integrated, like internalized? What will happen? 是个拔河。
所以就是看你 target 选择，像 PD-1 的 target， 你把它即使你还来不及 engage 到 T cell， 那你 reduce PD-1 的 expression，I think that's a good thing. Same as 你 HER2 variant in the case of AP402。可是 in reality， 我想你如果你 infiltrate 到 tumor 里面 antibody 的量够多。或者说它先办到 CD37， 那基本上被target cancer target internalize 的机会应该就不大了，所以这些到最后还是要做实验去做验证。
When we design a bispecific antibody, we have to face sink effect. It means you have to balance the affinity between two targets, right? So in your design, how do you avoid this kind of problem? You know, if you bind one target with very high affinity, you won't have a chance to grab the other target, right? So how do you balance this kind of hurdle?
我们的 anti-CD137 的 affinity 大概在2 to 3 nanomolar range。我们的 cancer targeting antibody affinity 基本上都在 sub-nanomolar， 所以事实上的确它对cancer 上面的 affinity 其实是比较强的。那会不会造成问题？那至少在我们cell-based functional assays 跟我们的 tumor model 上面。目前我们没有看到有什么太大的问题。其实一个相关的问题是，其实很多人问这个问题，就是你同时办到cancer cell 上面跟 T cell 上面的时候，到底在 molecular level 你是怎么去 activate T cell？ 这个其实是很多人在问的。那我简单的说好了，就像我刚刚提到的，在 NK cell 或是 T cell 上面，当你 engage 到你的 target cell， 比如 bacteria， 甚至 in this case cancer cell 的时候，当你有足够多的 binding event， 你事实上可以形成一个所谓的 immune synapse。Okay？ 在这个情况之下，你的target cell killing 的效果就会特别好。我们的 bispecific antibody in a way behaves like that theoretically. Yeah.
Okay, we'll move on to the last presentation of this morning before our lunch break. Dr. Jen, Dr. thank you very much for your presentation. 谢谢，谢谢。The next presentation should be very interesting for many people here and for the audience, from Taiwan or from, especially from the biotech communities of Taiwan, because CDMO is considered to be a major direction for us to develop biotech industry in Taiwan. And this next presentation is about CDMO, the business of CDMO, the technology used in CDMO, especially with biologicals. Dr. Wei Chang Zhou is from 对岸我们的朋友从对岸过来他是 Dr. Zhou is at the very top of the company, WuXi Biotech. He's sitting on the board for many years and still on the board. I think he himself is an expert in various technologies used in the manufacturing, the characterization, the other aspects of biologicals. WuXi Biologics is known to be a leader in the field of CDMO in terms of the diversity or the scope of the technology used by the company. I think they are the foremost in the industry. I won't talk about these technologies. He will explain to us. So let's welcome Dr. Zhou.
Okay, I guess it's always a dilemma for me to decide speaking Chinese or English. It's also before the lunch, right? And it's a little late, so I probably will speak mixed. And we'll use English because I was told that the conference should be in English. So all the slides are prepared in English, so there's no Chinese. But seriously, first of all, really thank you for the invitation to be here in Taiwan. And I have had great interactions with scientists and companies in Taiwan for many years. And I remember gave the first talk here while I was at the PDL Biopharma, which is an antibody discovery company. That was many years ago. Had a really great tour. And then while with WuXi Biologics, I was here 2016 to attend the Bio Taiwan, and they introduced WuXi Bio. But I think the more significant I want to say is also that even though I have worked many in China the last 12 years and before was in US, but I really have a very close tie with Taiwanese companies. I think particularly Dr. Zhang, and we work with Zhongyu Xinyao and TaiMed, bringing the anti-CD4 antibodies and literally from early stage to the BLA approval in 2018, first by FDA, then by EMA. I believe this may be first innovative antibody which discovered in Taiwan and then enabled by WuXi Biologics now, which benefits the patient worldwide for HIV patient. So what I actually want to talk about today is really talk about some innovative technology we haven't been working on really for last 12 years since I joined WuXi Biologics. And in between, probably I will just also give you some ideas about what we have done to enable global biologics development and manufacturing. So clearly, I think this is a quick disclaimer because we are a public company. So really, I would probably very briefly talk about emerging trend, challenge, and opportunities. Both Professor An and Dr. He give a very good overview about really the trend of antibody therapeutics, including monoclonal bispecific, and then obviously ADCs mixture. Then I probably will focus on really how we can get efficient and bring an idea, bring on what we can have discovered, for example, in Professor An's lab, how to bring that quickly to the IND, to the clinical trial, then also to the final approval without spending $1 billion US for the development. I think that's the challenge we all face. We all know that there are so many ideas and in our laboratory worldwide, but how to bring that to patient quickly and develop in an efficient way. And finally, if I have time, we'll talk a little bit the effort we have put for the digitalization, automation, to enable more efficient development and manufacturing. So this is I can quickly go through, which Dr. He gave a very significant overview. But I think the take-home message is that the US FDA, for last, I would say, 15 years, proved about 20% of the new molecule, they are biologics. So essentially, biologics haven't been and part of the therapeutics and will be staying. And in addition, this figure also, Dr. He maybe mentioned before, is from monoclonal antibody, now go to plus. The plus, I think the first is bispecific antibody, then is a combination of bispecific and with ADC. And so that's an immediate trend. But I think there's also many more recombinant proteins, fusion proteins. And I think if you look at a blockbuster, and I think I would not belabor this clearly, that why we have such big industry for the last 30 years is because all these products, they are help patients. By helping patients, you actually follow, you get the sales of the, essentially the blockbuster sales. Now, if you look in and further in terms of technology for the product-wise, the maturity clearly is from a very small molecule at the beginning and mature technology. And now we are in a stage like a bispecific antibody, ADC, but particularly, and which I will not talk about, but I think gene therapy, mRNA-based therapeutics became emergent, much more advanced trend. Then obviously there's much more, like the cell therapy, and which is a lot still in the research, but not in the commercialization. So if you look in the product modality, antibody is here to stay, but we add it, so-called antibody plus, then cell therapy and the gene therapy, mRNA, then even microbiome and product. So by doing that, why we created so much modality, what needed is technology to enable those faster, bring those medicine to a clinic. So what I will spend my time is really talk about what are key challenges for biologics development and manufacturing, and what we as a CRDMO and part of the ecosystem, how we contribute to these to solve these problems. And clearly, we all know very well, and that's what we all want to do. So strategy-wise, it's very simple in a way. But in order to achieve that, it's very difficult. I mean, we all know we need to put the quality first, right? Nobody wants to compromise the quality. And the second is that you want to be fast. to get to clinic, get to conventional, right? Then at the cost you can afford. So that's traditionally we all talk about the triangle. If you can get a 2 of 3 right, you cannot get a third right. But we actually did one more, which is flexibility for all these modalities. So really for the last 12 years, what we have done is really we call C and C is build the capability and build the capacity so we can help and enable this development to reduce the timeline from DNA to IND, then to either EUA and BLA, then increase productivity to reduce cost, then ensure consistent high product quality, then obviously improve development efficiency. So that's having been the challenge for us since day one, and will be continue the challenge for us for the next 10 years. We all have to do that. So in order to do that, what we have done, and this may be a quick summary of our biologics development technology and capability. So over the last 12 years or so, WuXi Biologics, we actually built the leading technology platform. First, to support discovery research, we use CHOK1 cell line to support customized protein production. Then move to cell line development. Now we have CHOK1 cell line platform, also ADCC enhanced cell line platform. Then move to processing. We have so-called continuous bioprocessing platform, WuXi Up. Then also for fed batch, we have ultra-intensified fed batch platform, which can get us somewhere 35 grams per liter for antibody. And so that's very high productivity. Then obviously, we also need to build for the high concentration, for the delivery of the product, for the formulation, then build the, and one of we haven't talked much is the analytic to make sure we can characterize, release the product efficiently. So we actually build 5 analytic we call center of excellence, from bioassay to mass spec to forensic and the process impurity. Then also the drug product center of excellence to enable delivery of product. So essentially, we have technology cover from cell line to the drug product. So the major service scope we support is protein science, both also synthetic biology. And we have focused on mainly on mammalian cell expressed biologics, including all antibodies, recombinant protein based on mammalian cells, and then clinical manufacturing and the release, then conventional manufacturing. But we also have with so-called biosafety testing in-house, and this will enable very fast product release to the patient. Then for last a few years, we built a new platform based on microbial, mRNA, and the viral platform for the other product modality. And obviously, in order to do all this, we have to have a good CMC strategy and project management. So we should do everything right from A to Z, not just one aspect. So just on the development side, now we have a team about 5,000 people globally. WuXi Biologics total now have 12,000 people, so about 5,000 dedicated for development. Those are based on in US and in New Jersey, Pennsylvania, and also in China. And as we speak, we have built a center in Singapore to enable development in Singapore for both development and conventional manufacturing. So as a result of technology of our team, so we actually bring the timeline, like for biologics from DNA to IND, within 10 months can get, once they give us DNA sequence, we get your material for IND approval. And then give it to IND for BLA within 15 months. So we have done that. And probably you all know that I don't need to mention that we are unable, for example, COVID-19 antibody. From the day we get the DNA sequence to US approval within 14 months. And the following 10 months, we provided about 2,000 kilograms of the antibody to US market. And so that's a very fast speed. And overall, our track record right now, worldwide, we support 698 integral CMC projects. This including more than 100 projects by specific, and Dr. He talked different challenges. We have seen all of those, and I will not need to repeat. Also about 140 ADC projects, including the 700 projects. And last, I would say 6 years or so, we are able to close to 500 IND approval worldwide. So that's a very large number because just give you an example, the leading company in biotech, like Amgen or Pfizer, they probably may have 10 IND approval a year. And so we actually are able more than hundreds INDs a year approval. So that's a very large number. So I think we work with 600 global partners, and within our short history, we supported 33 market authorization approval, including the time-out product. So how we get that? And I mentioned the speed. This obviously, and we cannot get here from one day. So clearly, the first I want to talk about kind of the speed in terms of fast biologics development. Back when 2015, not many years ago, and from DNA to IND, which would take us 24 months. Then we came up with technology, reduce this timeline every 3 months until 2020, which is 12 months. Now we routinely provide from DNA to IND within 10 months. Part of this is driven by really by infectious diseases. And at the beginning, we work on Zika virus antibody, yellow fever antibody, reduce this timeline until we get COVID, which is 2019. We reduce the IND timeline literally within 3 months. And in addition, while we work on the platform from DNA to IND, we also obviously work on the activity from IND to BLA from typical 2 to 3 years to 15 months. So that's enable us to bring the product faster to clinic than to commercialization. And we probably haven't even thought about it 10 years ago, and we will be able to support 33 market authorization within 10 years. So that means 3 product approval a year. And here, I think in terms of bispecific, I want to just briefly, and bispecific is quite new. Back 5 years ago, 6 years ago, we only work on 10 projects. And last year, this number increased to 114. And the timeline from the bispecifics also reduced dramatically. Now, for typical bispecifics, we can deliver IND within a year, 12 months. And so far, we are unable to 60 IND approval for bispecifics. If you look in the pipeline, so mostly still in a preclinic, but also moving now to later stage, phase 2, phase 3. So we expect that some bispecific are enabled by WuXi Biologics will be on market pretty soon. And those some of the technology, and I know we probably don't have the time to talk in this audience, but I also want to highlight that we haven't shared this knowledge with community internationally. So this is 3 papers we started published 2021, and this is also 2024, and here is, and also recently, and 2022, we published how we shape the cell line development CMC strategy and for COVID antibody, and then actually applied this to long COVID program, which just published and now. So if you are interested in clearly, and we can discuss offline about some of the details, how we get in there. But in fact, we haven't been there. So some of the key highlight I want to show technically is really our cell line system. We based on Chao-1 cell line system, and we came up with this platform called WuXia platform. And then later we also have the ADCC enhanced. To summarize some of the data, like for use this cell line platform like monoclonal antibody, which much more predictable and the productivity somewhere from 4 gram to 10 gram per liter. For fusion protein, and maybe a little lower, but for bispecific, especially for 80 days, the bispecific antibody titer is really low because everyone have their own platform, and now we can get also pretty high at the 10 gram per liter per day. And Dr. mentioned not just the titer, it's also the product stability and the product monomer. So, and in addition, for cell line platform now, we also have a HEC293 cells for some special recombinant protein. So not just CHOW cells, also the 293 cells. And I mentioned in terms of productivity, and in the interest of time, I want to highlight our we call ultra-intensified fed batch bioprocessing platform. So here we actually can, from typical fed batch, and like 4 to 10 gram per liter, we can increase the additional 3 to 6-fold, so reach about 10 to 25 gram per liter. And in some extreme case, we reach the 35 gram per liter. And if you look in some project we work with, this is about 15 projects, and show from the titer and the typical fed batch to this ultra-intensified fed batch, we have fold increase somewhere from 2-fold to about 5-fold of increase, and for most projects. So this actually, we don't need to have such high productivity for every product. But if you do have a product which have a very large demand, like Alzheimer antibody, like this and check inhibitor and antibody, you need a large amount. We actually can use a single-use bioreactor. We call use scale-out strategy, just by use multiple single-use reactor, then reach high titer, reduce of the cost of goods. So that's our strategy, our technology. And if you also further by team up our, we call WuXi ultra-intensified with our in-house media, and we actually can see for different cell line, we can see further improvement. The marketing chow is the media, in-house media with commercially available media. We can further improve the productivity. And from typical fed batch, for example, from commercial media to our in-house media, there's improvement. Then intensified fed batch go to 15 gram per liter, also improvement. Then for the ultra-intensified, I mentioned 35 gram per liter. So, and obviously we cannot improve every to this such high titer, but you can see literally this applied for many, many projects, and we see the systematic improvement. And another thing would be, and I think this is kind of dear to my heart, is that since I worked on continuous process via the Genzyme, now it's part of Sanofi, and it really came up a new technology we call continuous bioprocessing, improve productivity. So here at the beginning, we was either upstream, use so-called fed batch culture, and also intensified perfusion culture to improve productivity on the upstream. But the last few years, what we really have worked on is trying to implement both upstream and also the downstream fully continuously. And so essentially, now we are able to, from a vial to the final drug substance, we can have a fully continuous processing if you need. So this further improves the efficiency and the productivity. And I believe as one example, I probably want to talk about highlights that in one of the cases that this was implemented last year, we implemented a non-GMP pilot plan at the 50-liter scale. And so this 50-liter scale, we actually can produce 3 kilos of the drug substance within 25 days. The total productivity over 25 days is 105 grams. Downstream yield is 80% with very high product quality and both monomer and it's very high. And then I think the cost of goods, even at 50-liter scale, can reduce to $65 per gram. So that's some of the data you can see. Obviously, this we now implement in many other projects as well. So by doing that, we can dramatically reduce cost of goods. So here is over the time, I believe is 70 some project, and for the 136 different clone, we for bispecifics, fusion protein, monoclonal antibody, recombinant protein, we actually fully implement this now. Now you have the productivity. In many cases, would be 4 to 6-fold of improvement. And I mentioned that we successfully implemented this continuous processing and both upstream and downstream, then at the pilot scale, 6 grams per liter per day. So we actually published this as well. So one of the key things, and I want to spend a couple minutes to talk about, is really process analytic technology. When we do continuous process, the concept is very easy. Everyone knows how to do it. But in order to implement that reliably, not many people can do it. I can say that. Many people knows how to do it, but they cannot do it. So this is always different. So we actually spend a lot of time over the year to build a robust process analytic technology. And here's a kind of from upstream and intensified perfusion, then go to multiple column continuous product capture, then online and viral inactivation, and some of the downstream purification. Now for the upstream, now we implement the so-called Raman spectroscopy for titer testing, as well as for metabolites and cell culture testing. For downstream, and we have some limited implementation of inline pH, tubility, online HPLC, and inline concentration flow through, etc. So this would add more and more process analytic technology to enable automatic continuous processing. So the example for the Raman spectroscopy application, this concept is not new. Raman hasn't been used for mammalian cell culture maybe for 20 years, but no one have taken that to a degree. So essentially what we are doing is that use just one Raman probe, then based on the Raman analysis, then through a software, which updated the software, then we can get a number of the parameters, including we all know metabolites, lactate amino acid, osmolality, and cell culture, cell-related parameter, titer, and then we also now to use Raman to do product quality attribute like purity, charge variance. So what does this mean? Essentially, one measurement, they can replace all the measurement now in your lab. Imagine in 5 years, all the equipment, all the instrument you buy today for QC is not needed. And this is not just save capex, improve quality, but also save a lot of manpowers. So reduce the cost. We applied this now to about 20 projects, both in benchtop, then also large scale up to 2,000 liter. And that's the one, and I reflected myself, why this now became this is so reliable, partly is due to the computer, due to the AI, due to the digitalization. Now we have much more database, and the biotech industry is converging to a very simple platform. You have CHO cells, serum-free media. So the only difference is the product. Everything else is very similar. So you can rely on what is the data you generate from the previous project for the future. And then because of AI, because of mathematical modeling, so they're actually learning better than human. And I was fortunate to recruit a lot of smart scientists that work in the lab. So we are able to make this work and publish also significantly. And again, I mentioned the new advancement, the PAT, process analytic technology for biologics. So PAT, this would be for media analysis. And when I was myself down in the lab, you always done sample prep, detection. Now all this can be done online. Then further is that so-called MAM. More efficient multiple attribute method now use MS. It's not just you measure one parameter at a time, you actually measure multiple parameters at the same time. So this again improve efficient and improve the way how we know biologics. And in addition, I think one sense probably Dr. He did not talk about is for bispecific, multi-specific, is the bioassay. It's not just the binding assay, it's also the cell-based assay. When you talk about 1 + 1 is not equals 2, this will be more than 2 in terms of bioassay because you have 2 different targets, you need to have bioassay. Then you have a combination. And so the bioassay, which we built a very large team to do the bioassay, and as well as for the mass spectral toolbox for the product quality analysis. So going forward, what we will do is that not just doing the cell-based and binding assay, is also use real-time determination of biokinetics for biologic with a target molecule. This will be used is a service plasma resonance, SPR, BLA, etc. Those methods. So we think those technology would revolutionize the QC for biologics manufacturing. So finally, I want to show you maybe one video to see what is the automation and we are doing in our lab, and we hope this first improvement in the manufacturing. So this is one of robotic we have built it for few years now. Is implemented. They can take sample automatically. We call this now in dark lab. So in our lab, we don't even now see any scientist. So I think that this robotic takes the sample and prepare the sample, fit into traditional analyze to count the cells, and also measure tighter product quality. So this is became a reality. So the next step, what we kind of really do, we already prioritize this smart clinical manufacturing platform based on the automation, and this would definitely would be automatic feeding, then the drug substance allocation, then the ultrafiltration, diafiltration, and sampling. So we have done that. And I show you some example of process analytic technology based on Raman and also based on DS concentration measurement. So we are further to push this boundary for digitalization, use Pi deployment for data capturing, and this then later, hopefully use this data, we have much more mathematical modeling, bioprocess modeling to get we call smarter manufacturing and going forward. So we believe this will be the future. So finally, and I don't know if I still have time on that, I will just quickly summarize. So over the last 10 years, we have relentless focus on how to drive the speed and for biologics development, then reduce the cost by improve the titer, improve the efficiency for manufacturing. And obviously all this is easily said and done. So really what we build is a foundation for the new technology for cell line, bioprocess, analytical formulation to accelerate the development, reduce cost of goods, ensure the quality. And I believe there's a lot to be done, particularly in the digitalization, automation to enable transition of current development and manufacturing to a much more efficient and better and smarter the future development and manufacturing. So within that, I want to thank definitely my team and also partners which enable us to use this technology and to move their project faster to clinical and to commercialization. So thank you for your attention, and I will be happy to answer any questions you may have. Thank you.
Thank you. Thank you, Dr. Zhou. Dr. Zhou just gave us a glimpse of the enormous capability WuXi Biologics has in doing this business in biological process development, manufacturing, and so on. Before we open up to the audience for questions, may I ask you a question? And I think the audience will also probably have the same questions. How do you compare to to say the other big CDMO companies like Lonza or Samsung Biologics, in what you do in technology-wise or business-wise, how you are different among, say, the three of you?
Sure. I think a traditional business is more so-called CMO, right? So both Samsung and Lonza and have focus on larger scale manufacturing and to make that efficient and make that available to everyone to use. And WuXi Biologics actually came to a really good timing. So we started to link in discovery and development to commercial manufacturing. So essentially, we have been focused on so-called strategy we call follow the molecule strategy. So essentially, we are not just focused on how to help you manufacture. We're trying to team up with our client, our partner from discovery, development to commercial manufacturing. So that's what we have done last 10, 12 years. So we call follow the molecule. Obviously, when you are doing very good, then we can say, win the molecule. Then we can have the project transfer to us at the early stage of your program. So that's probably the major difference. So we actually call us, and we probably the first company to call us a CDMO. Now we call us a CRDMO. So that's probably the major difference. But I think eventually everyone is learning, everyone is getting together. So I know, and other companies is also trying to build a development capability.
Okay, thank you. Yes.
Yes, Richard, that's a beautiful talk. So about 20 years ago, the cost of good for antibody is about $200 per gram. So you reached to about average $50, $60 per gram. Do you think you reached the limit for the cost of good, or you can continue to improve with AI or the technology you're talking about?
Yeah. I mean, I think just take a step back. I mean, for antibody, when I first, myself was a lab scientist, I remember at the Merck days, right? We was making antibody at about $10,000 a gram. And so this kind of make the antibody not feasible for large-scale commercial manufacturing. So this is the same today, like gene therapy, CAR T, which I think is a million. However, for the last 10 years, we definitely reduced the cost to more than 2 log, from 10,000 to 100, right? So if gene therapy, CAR T therapy technology can enable 2 log reduction, I think gene therapy, cell therapy would be a good in business as well. But to go back to the biologics, I think what would be limit? Current technology, I think, can get us somewhere $25 to $50 per gram at a reasonable amount. But if you really have very large scale manufacturing, you probably can further. I mean, some people project that this can go down to $10 per gram. I think it's feasible, but you do need a very large scale. I will mention that. We work many on project. Some of for like rare diseases, you don't need so much, right? But for the Alzheimer's disease, you need a lot because the berry. So I think it can go down to quite a bit.
The presentation is very amazing that how you have progress with that all the using the PET. Especially for online, all the measurements. I have the questions are really regarding this geopolitics regarding the Biosecurity Act in 2024. So I don't know that whether WuXi or the senior management has any action to take on this one. Yeah.
Yeah, I mean, I probably not expect the question, but thanks for the question. And obviously, this is on everyone's radar screen, both in the US as well as back in Asia, including Taiwan and mainland China. We all talk about that. I think what this clearly, and what we can do as management, and I'm no longer active management, I'm the director of WuXi Biologics. I think what the advice I will give to current management is, which they are doing, is really focus on what we can do. I mean, what today we are doing, I mean, as I show you, is really complementary to any company that may need this technology. And I think, I mean, yesterday I had a delayed flight from San Francisco to here, and I had an opportunity to learn this first time in my life. I know Bitcoin, but I did not know crypto, right, the technology. And there was an interview about this Bitcoin and how Bitcoin business would be good investment. I never invested that, and now I really regret it, right? Because Bitcoin, 10 years ago, probably is about $100 a piece. At the peak, it was $70,000. Even now, it's $30,000. So if say people invest in Bitcoin 10 years ago, you will be rich man today. So, what I'm saying is that I know there will be a lot of discussion about Biosecurity Act, and every country, every politics will talk about that. But I think from the recent, first of all, that's still a draft, right? Still not passed into the law. And the second is that just recent draft had also significant amendment. So what we really focus on is to relentlessly reduce the cost to get the high quality and fast, so we can help these companies. And by help the company is help the patient. And my first job is within Merck. So I stayed at Merck for 8 years. And I think Merck brainwashed me that medicines for people, not for money, right? That's Merck still saying that after. And I did not, at that time when I was young, I thought Merck may not survive for very long. But Merck still is one of the largest pharmaceutical company doing good things for patients worldwide. So I think I probably would just follow that, continue follow that, really provide a value to our client, to our partner, wherever they are.
Do we allow one more question?
Very impressive, the capacity and the efficiency. You mentioned that from DNA to IND that you can achieve within 10 months. So because for the IND, excuse me, for the IND application that is required GLP tox study. So I'm wondering that how you are able to facilitate the GLP tox study to achieve the IND application within 10 months.
Yeah, I think for most GLP tox study, including report, take about 6 months. So it's 3 to 6 months. So we work with them very closely. Essentially, we provide the material at the time like 3 months, 4 months for the beginning to do that. But we also, I mean, I don't know, I mean, there may be a lot of people here that are doing GLP talks. So I don't want to pressure them. But there's also room to reduce that timeline. I think the animal, obviously, in lifetime, probably you cannot reduce much. But I think the testing, reporting, which would take a long time, I think that's kind of rate limited step. The COVID, why we can do so fast, is COVID is a neutralization antibody. The tox study is less than a month. So that's how you can. CMC, I believe we reached quite a limit. We can do it within 6 months. In fact, I did not mention during the talk that we actually helped many companies outside of China, and for the oncological antibody for cancer, and mostly it's 6 to 7 months. Because for some cancer antibody, you don't need a long GLP study. But for autoimmune diseases, multiple dose, that's a different story. Yeah, you probably cannot reduce the timeline even further.
Is there any burning question you want to ask? Last one. If not, we'll stop here for lunch break. Thank you very much, Dr. Zhou.
Okay, we thank all the speakers and moderators of this morning. So it is lunchtime now, so for our VIP guests, please proceed to the VIP room on the second floor for lunch. And for other guests, please go to the third floor to pick up your lunchbox with your badge. And we also have a lunch seminar on the third floor, which is hosted by our diamond sponsors, Bosen, Baysiter, and Brooker. So please also attend the lunch seminar. Thank you, and please come back at 1:30 this afternoon. Thank you.
details, a quick reminder that today the pharmaceutical industry is facing a real challenge where the cost of developing new drugs is now so high that the rate of return on investment is soon going to be negative. And to reverse this trend, we're going to need new modalities of drugs to increase the number of drugs. And we also will need to increase the efficacy with which we identify new drug targets, and to make sure that we're working on target that will have the highest likelihood of being successful in a very cost-intensive stage of clinical trials. So we need to increase the efficiency with which we identify safe and effective new drug against those targets. And finally, we need to increase the quality and expected commercial value of this drug. And hopefully that's where we can help. So how can Twist help address those challenges? Well, we believe that our platform that miniaturizes the chemistry of writing ACGT on our silicon chip maximize the throughput. We can make millions of oligos every day and lower the cost of DNA, which can now unlock a number of applications. So let's start with target identification. The last decade I've seen some incredible breakthrough in our ability to discover novel targets for treating disease. And we can't talk about that target identification without talking about CRISPR and how genome-wide CRISPR screening has enabled finding genetic elements that are linked to phenotypes. And thanks to the millions of oligos that Twist makes, now very high quality data can be produced. And here in the example published in 2019 by the Sanger Institute in Cambridge, they've shown that screening with Twist oligo pools of 324 human cancer cells of 30 different types resulted in really high quality data and a number of candidates for downstream cancer therapeutic. But now scientists are able to move beyond just silencing one region by using dual guides. And that creates a bunch of new applications in functional genomic screening. For instance, you can excise promoters, you can excise link RNA, you can excise non-coding elements, or you can actually look at systematic interaction of one gene pair with another gene pair. And so the barrier to entry to be able to do that is actually the length of a dual guide. A dual guide is beyond the ability of normal DNA synthesis, but at Twist we have addressed that challenge by being able to make 300 base pair oligos. And so that means that now scientists can fit two single guide RNA together. They can fit a Cas12a with a CR-RNA array and basically expand the design space that they have. And so now we're enabling combinatorial screens without having to worry about the downstream cloning issues, without to worry about the cost. We can take care of all of it at a very cost-effective manner. And as an example, we are now seeing a lot of great publication in the literature using those dual guide scanning. So now that we've done the target identification, we can go into the drug discovery and development. And we believe that Twist has the potential to accelerate biopharmaceutical and immunotherapy development through rapid antibody optimization and humanization. And that's super important because in 2019, 4 out of the 5 best-selling drugs were biological. So that ability to discover and optimize biological drugs is key. However, it's inherently complex to optimize biological drugs, and the development takes time. And so at Twist, we offer scientists a toolbox to enable them to quickly find new and efficacious leads against targets or even against hard-to-drug target, which means, and then we give them the means to very quickly optimize drug-like properties for those molecules. And as an example, at Twist, we offer two things. Starting at the bottom is our hard-to-drug target discovery. using our libraries that are inspired by the human repertoire, we've been able to show that we can very effectively pan those libraries and find leads even against hard-to-drug targets. And then at the top, if you already have an antibody, we're able to optimize an antibody against using the power of our libraries. We can design in the computer massive libraries, synthesize them, and then bind them to find either biobetter or finding molecules that have better drug-like properties. And we've done that at Twist for many different targets. We've done GLP-1R, we've done a number of targets in immuno-oncology like PD-1, TIGIT, CD3, ADORA2A, and we also have done it against COVID-19. On the basis of those great data, we've been able to sign a number of fantastic partners in many different modalities, in many different indications. And if we look at the statistics of what we've achieved at Twist, we've completed 19 different programs, and we still have 43 programs that are active with 31 partners. And again, the indications can be very broad, from cancer to neuro to immuno-oncology, infectious disease. We even do drug discovery and optimization for cats and dogs. And then we have very varied modalities, starting from VHS, antibody, bispecific, ADC, TCR, and more. And as I mentioned, but the root to achieve those results is our ability to make amazing libraries. Because we have a silicon chip where we can make millions of oligos at the same time, we can create explicitly millions of genetic elements that have any mutations that you want. And then those elements are super precise. So it's a base by base precision. It's a codon usage precision. We can also precisely do combinatorial assembly of those elements. We can control super precisely the ratio of amino acids. We can control the length of the domains, the length of the CDRs. We can remove restrictions side. We can remove unwanted motifs. We can also pop in that genetic material into many different scaffolds so that you have combinatorial of CDRs, but also you have combinatorial of scaffold. And then at the end, everything is NGS sequence so that you know that the quality is there. So again, that is at the root, and that makes sure that you have basically a synthetic library that is human in content. So it's a semi-synthetic library, which is the best of both worlds. And if we contrast those libraries against other libraries like NNS, NSK, or TRIM, we are the only one that can match the natural human repertoire and that can remove the liabilities from the libraries. So now that we've talked about discovery and optimization, we can very briefly talk everything that we do. From target identification to target validation to drug discovery to drug development and even to disease stratification. We have the tools at Twist from our genes, oligo pools libraries, our NGS kits, and our biopharma products. We have the tools to enable you, help you get to your answer. And so we look forward at Twist to working with you and enabling you to get through those stages and write the future together. Thank you very much.
CARTERA LSA platform is a revolutionary and versatile, high-throughput antibody discovery platform that combines patented continuous flow microfluidics with high-throughput surface plasma and resonance to deliver up to 100 times the data in 10% of the time and with only 1% of the sample requirements compared to other systems. The LSA enables you to automate key applications in single unattended runs, including capture-based kinetics assays and epitope binning for interrogating up to 384 by 384 pairwise interactions. The single flow cell sample needle can access a sample block position with various tube formats up to 50 milliliters and a microplate position which can hold a 96 or 384 well plate. The 96-channel printhead can immobilize up to 384 distinct ligands to the array. Vertical, bidirectional flow in the flow cell enables extended contact times during immobilization to concentrate material onto the surface of the array. The single flow cell covers the entire array, enabling a simultaneous analysis of up to 384 binding interactions. The LSA can collect fully referenced kinetic data for 384 clones simultaneously. Industry-leading data analysis software enables kinetic characterization and epitope binning analysis in minutes. The CARTERA LSA software features an intuitive user interface designed to help you set up your experiments quickly and efficiently. With epitope binning software, data are linked across panels to facilitate review of sensorgrams, heat maps, networks, and a dendrogram. The ability to merge orthogonal data and view networks by color options provides a powerful and intuitive way of assessing your antibody panel's diversity while keeping an epitope-centric view. To learn more about the CARTERA LSA and how you can use it to generate 100 times the data in 10% of the time, using only 1% of the sample compared to other systems, visit us online at cartera-bio.com.
Hi, my name is Emily Liprus. I'm the CEO of Twist Bioscience and one of the 3 co-founders. I'm very happy to talk to you about writing the future of drug discovery and development today. So first, I'll start by saying that I will be making some forward-looking statement. And I'm super excited because DNA is changing for the world for the better. That's what we do. We write DNA from scratch, and we want to help you solve the many critical challenges that humanity faces. The focus today is to focus on drug discovery and development, where synthetic DNA has a potential to rapidly drive improvement in our ability to treat diseases. So before we go into the details, a quick reminder that today the pharmaceutical industry is facing a real challenge where the cost of developing new drugs is now so high that the rate of return on investment is soon going to be negative. And to reverse this trend, we're going to need new modalities of drugs to increase the number of drugs. And we also will need to increase the efficacy with which we identify new drug targets, and to make sure that we're working on target that will have the highest likelihood of being successful in a very cost-intensive stage of clinical trials. So we need to increase the efficiency with which we identify safe and effective new drug against those targets. And finally, we need to increase the quality and expected commercial value of this drug. And hopefully that's where we can help. So how can Twist help address those challenges? Well, we believe that our platform that miniaturizes the chemistry of writing ACGT on our silicon chip maximize the throughput. We can make millions of oligos every day and lower the cost of DNA, which can now unlock a number of applications. So let's start with target identification. The last decade have seen some incredible breakthrough in our ability to discover novel targets for treating disease. And we can't talk about that target identification without talking about CRISPR and how genome-wide CRISPR screening has enabled finding genetic elements that are linked to phenotypes. And thanks to the millions of oligos that Twist makes, now very high quality data can be produced. And here in the example published in 2019 by the Sanger Institute in Cambridge, they've shown that screening with Twist oligo pools of 324 human cancer cells of 30 different types resulted in really high quality data and a number of candidates for downstream cancer therapeutic. But now scientists are able to move beyond just silencing one region by using dual guides. And that creates a bunch of new applications in functional genomic screening. For instance, you can excise promoters, you can excise link RNA, you can excise non-coding elements, or you can actually look at systematic interaction of one gene pair with another gene pair. And so the barrier to entry to be able to do that is actually the length of a dual guide. A dual guide is beyond the ability of normal DNA synthesis, but at Twist we have addressed that challenge by being able to make 300 base pair oligos. And so that means that now scientists can fit two single guide RNA together. They can fit a Cas12a with a CR-RNA array and basically expand the design space that they have. And so now we're enabling combinatorial screens without having to worry about the downstream cloning issues, without to worry about the cost. We can take care of all of it at a very cost-effective manner. And as an example, we are now seeing a lot of great publication in the literature using those dual guide scanning. So now that we've done the target identification, we can go into the drug discovery and development. And we believe that Twist has the potential to accelerate biopharmaceutical and immunotherapy development through rapid antibody optimization and humanization. And that's super important because in 2019, 4 out of the 5 best-selling drugs were biological. So that ability to discover and optimize biological drugs is key. However, it's inherently complex to optimize biological drugs, and the development takes time. And so at Twist, we offer scientists a toolbox to enable them to quickly find new and efficacious leads against targets or even against hard-to-drug target, which means, and then we give them the means to very quickly optimize drug-like properties for those molecules. And as an example, at Twist we offer two things. Starting at the bottom is our hard-to-drug target discovery, using our libraries that are inspired by the human repertoire. We've been able to show that we can very effectively pan those libraries and find leads even against hard-to-drug targets. And then at the top, if you already have an antibody, we're able to optimize an antibody against using the power of our libraries. We can design in the computer massive libraries, synthesize them, and then pan them to find either better or finding molecules that have better drug-like properties. And we've done that at Twist for many different targets. We've done GLP-1R, we've done a number of targets in immuno-oncology like PD-1, TIGIT, CD3, ADORA2A, and we also have done it against COVID-19. On the basis of those great data, we've been able to sign a number of fantastic partners in many different modalities, in many different indications. And if we look at the statistics of what we've achieved at Twist, we've completed 19 different programs, and we still have 43 programs that are active with 31 partners. And again, the indications can be very broad, from cancer to neuro to immuno-oncology, infectious disease. We even do drug discovery and optimization for cats and dogs. And then we have very varied modalities, starting from VHS, antibody, bispecific, ADC, TCR, and more. And as I mentioned, but the root to achieve those results is our ability to make amazing libraries. Because we have a silicon chip where we can make millions of oligos at the same time, we can create explicitly millions of genetic elements that have any mutations that you want. And then those elements are super precise. So it's a base by base precision. It's a codon usage precision. We can also precisely do combinatorial assembly of those elements. We can control super precisely the ratio of amino acids. We can control the length of the domains, the length of the CDRs. We can remove restrictions side. We can remove unwanted motifs. We can also pop in that genetic material into many different scaffolds, so that you have combinatorial of CDRs, but also you have combinatorial of scaffold. And then at the end, everything is NGS sequence, so that you know that the quality is there. So again, that is at the root, and that makes sure that you have basically a synthetic library that is human in content. So it's a semi-synthetic library, which is the best of both worlds. And if we contrast those libraries against other libraries like NNS, NSK, or TRIM, we are the only one that can match the natural human repertoire and that can remove the liabilities from the libraries. So now that we've talked about discovery and optimization, we can very briefly talk about everything that we do. From target identification to target validation to drug discovery to drug development and even to disease stratification, we have the tools at Twist from our genes, oligo pools libraries, our NGS kits, and our biopharma products. We have the tools to enable you, help you get to your answer. And so we look forward at Twist to working with you and enabling you to get through those stages and write the future together. Thank you very much.
CARTERA LSA platform is a revolutionary and versatile, high-throughput antibody discovery platform that combines patented continuous flow microfluidics with high-throughput surface plasma and resonance to deliver up to 100 times the data in 10% of the time and with only 1% of the sample requirements compared to other systems. The LSA enables you to automate key applications in single unattended runs, including capture-based kinetics assays and epitope binning for interrogating up to 384 by 384 pairwise interactions. The single flow cell sample needle can access a sample block position with various tube formats up to 50 milliliters and a microplate position which can hold a 96 or 384 well plate. The 96-channel printhead can immobilize up to 384 distinct ligands to the array. Vertical, bidirectional flow in the flow cell enables extended contact times during immobilization to concentrate material onto the surface of the array. The single flow cell covers the entire array, enabling a simultaneous analysis of up to 384 binding interactions. The LSA can collect fully referenced kinetic data for 384 clones simultaneously. Industry-leading data analysis software enables kinetic characterization and epitope binning analysis in minutes. The CARTERA LSA software features an intuitive user interface designed to help you set up your experiments quickly and efficiently. With epitope binning software, data are linked across panels to facilitate review of sensorgrams, heat maps, networks, and a dendrogram. The ability to merge orthogonal data and view networks by color options provides a powerful and intuitive way of assessing your antibody panel's diversity while keeping an epitope-centric view. To learn more about the CARTERA LSA and how you can use it to generate 100 times the data in 10% of the time, using only 1% of the sample compared to other systems, visit us online at cartera-bio.com.
Hi, my name is Emily Leproust. I'm the CEO of Twist Bioscience and one of the 3 co-founders. I'm very happy to talk to you about writing the future of drug discovery and development today. So first, I'll start by saying that I will be making some forward-looking statement. And I'm super excited because DNA is changing for the world for the better. That's what we do. We write DNA from scratch, and we want to help you solve the many critical challenges that humanity faces. The focus today is to focus on drug discovery and development, where synthetic DNA has a potential to rapidly drive improvement in our ability to treat diseases. So before we go into the details, a quick reminder that today the pharmaceutical industry is facing a real challenge where the cost of developing new drugs is now so high that the rate of return on investment is soon going to be negative. And to reverse this trend, we're going to need new modalities of drugs to increase the number of drugs. And we also will need to increase the efficacy with which we identify new drug targets, and to make sure that we're working on target that will have the highest likelihood of being successful in a very cost-intensive stage of clinical trials. So we need to increase the efficiency with which we identify safe and effective new drug against those targets. And finally, we need to increase the quality and expected commercial value of this drug. And hopefully that's where we can help. So how can Twist help address those challenges? Well, we believe that our platform that miniaturizes the chemistry of writing ACGT on our silicon chip maximize the throughput. We can make millions of oligos every day and lower the cost of DNA, which can now unlock a number of applications. So let's start with target identification. The last decade have seen some incredible breakthrough in our ability to discover novel targets for treating disease. And we can't talk about that target identification without talking about CRISPR and how genome-wide CRISPR screening has enabled finding genetic elements that are linked to phenotypes. And thanks to the millions of oligos that Twist makes, now very high quality data can be produced. And here in the example published in 2019 by the Sanger Institute in Cambridge, they've shown that screening with Twist oligo pools of 324 human cancer cells of 30 different types resulted in really high quality data and a number of candidates for downstream cancer therapeutic. But now scientists are able to move beyond just silencing one region by using dual guides. And that creates a bunch of new applications in functional genomic screening. For instance, you can excise promoters, you can excise link RNA, you can excise non-coding elements, or you can actually look at systematic interaction of one gene pair with another gene pair. And so the barrier to entry to be able to do that is actually the length of a dual guide. A dual guide is beyond the ability of normal DNA synthesis, but at Twist we have addressed that challenge by being able to make 300 base pair oligos. And so that means that now scientists can fit two single guide RNA together. They can fit a Cas12a with a CR-RNA array and basically expand the design space that they have. And so now we're enabling combinatorial screens without having to worry about the downstream cloning issues, without to worry about the cost. We can take care of all of it at a very cost-effective manner. And as an example, we are now seeing a lot of great publication in the literature using those dual guide scanning. So now that we've done the target identification, we can go into the drug discovery and development. And we believe that Twist has the potential to accelerate biopharmaceutical and immunotherapy development through rapid antibody optimization and humanization. And that's super important because in 2019, 4 out of the 5 best-selling drugs were biological. So that ability to discover and optimize biological drugs is key. However, it's inherently complex to optimize biological drugs, and the development takes time. And so at Twist, we offer scientists a toolbox to enable them to quickly find new and efficacious leads against targets or even against hard-to-drug target, which means, and then we give them the means to very quickly optimize drug-like properties for those molecules. And as an example, at Twist we offer two things. Starting at the bottom is our hard-to-drug target discovery, using our libraries that are inspired by the human repertoire. We've been able to show that we can very effectively pan those libraries and find leads even against hard-to-drug targets. And then at the top, if you already have an antibody, we're able to optimize an antibody against using the power of our libraries. We can design in the computer massive libraries, synthesize them, and then pan them to find either better or finding molecules that have better drug-like properties. And we've done that at Twist for many different targets. We've done GLP-1R, we've done a number of targets in immuno-oncology like PD-1, TIGIT, CD3, ADORA2A, and we also have done it against COVID-19. On the basis of those great data, we've been able to sign a number of fantastic partners in many different modalities, in many different indications. And if we look at the statistics of what we've achieved at Twist, we've completed 19 different programs, and we still have 43 programs that are active with 31 partners. And again, the indications can be very broad, from cancer to neuro to immuno-oncology, infectious disease. We even do drug discovery and optimization for cats and dogs. And then we have very varied modalities, starting from VHS, antibody, bispecific, ADC, TCR, and more. And as I mentioned, but the root to achieve those results is our ability to make amazing libraries, because
is an executive vice president of National Cheng Kung University. Please welcome Dr. Zhuang.
Good afternoon. In this session, we are going to have two distinguished speakers to give a talk about antibody development. Our first speaker is Dr. Haruhiko Kemada. I think he likes Osaka very much, so he got his bachelor degree, MS, and PhD degree from University of Osaka. And currently, he is the CSO of National Institute of Biomedical Innovation. So his work is very innovative. So today, his talk is technology to generate highly functional antibodies for antibody drug development. Let's welcome.
So thank you very much for inviting me to this great conference. And I would like to thank Professor Han-Chang Wu and Chuan for his kind introduction. So I work as the group leader in this National Institute in Japan, and also have been doing the research on the biopharmaceuticals using the antibody as a medical drug. So today, I would like to introduce our research on the technology to generate highly functional antibodies for antibody drug development. So in particular, I would like to show the result that the functional antibody depends on their epitopes and their association of the structure. So this slide shows the commercial sales of the biopharmaceuticals, and the global sales of biopharmaceuticals are $120 billion, making them a very huge market. And the global sales of the biopharmaceuticals, the top half of top selling drugs is biopharmaceutical based in showing this red panel. So as a result, the demands for the antibodies that can be used as a medical drug is increasing, and the technologies for using antibody as drug are attracting attention. So according to this chart, that shows the percentage of modalities of approved as drugs. So the small molecular drug is still dominant. However, the biopharmaceuticals using the antibody accounted for about a quarter. And in addition to the standard monoclonal antibody, the approved biologics is increasingly used a new format such as the antibody drug conjugate, and also the new modalities of the such as oligonucleotide. So the number of approved antibody drug is increasing every year, as is the number of the target of the antibody is increasing. So that's it used to be said that the number of target to discover was small, but in recent years, this problem has been almost solved. However, even though the number of the drug target is increasing, it can still be difficult to obtain the antibody that binds to the target molecule and shows the pharmacological effect. So this is a result of the flow cytoanalysis analysis to using the anti-PD-1 agent. So the binding to the PD-1 molecule is not so significantly different from the two medical commercially available antibodies such as the nivolumab and pembrolizumab, and also the antibody used for the experimental study. So how do you get the antibody with the desired activity in an efficient way? So let's consider the strategy for making the antibody to the receptor as a drug target. So how do you get the antagonistic antibody? So it can be imagined that the inhibition to the ligand and receptor is important for the antagonistic effect. So the ligand binding region should be target to make the antagonistic antibody. But how about the agonistic antibody? So the binding to the ligand seems to be important for the agonistic effect because the receptor can signal from the ligand binding. So the so the antibody should be target to the ligand binding region. But that is trouble because the function is completely opposite, but the target region is same. So I think that it is difficult to get the antibody with function image based on the image only. So this slide shows the difficulty of the making high functional antibody. So the binding of the antibody is the most commonly known feature of the function. However, it can be difficult to make the antibody. So, for example, the binding and bioactivity should be considered separately. So, in other words, the binding to the functional active antibody is not target the whole molecule, but rather targeting the region where it's function. So to make the functional antibody, it is necessary to identify the structural feature of the molecules rather than focusing on their binding to the target molecule. So the concept of the epitope is the important factor to understand the relationship between the function and their structure in the target molecule. So to make the functional antibody, we use our original method that calls the epitope normalized antibody panel, ENAP. So ENAP is the technology for generating the antibody to the target molecule epitopes to obtain the antibody panel, and that binds the epitope comprehensively. So to make the functional antibody, it is necessary to target to the function epitopes on the drug targets shown in here. However, using the typical method of antibody production is to make the antibody to each site where it's easy to make the antibody. So the screening based on the affinity and the frequency may miss this functional antibody because this method ignores the functional epitopes. So using the typical method, it is difficult to have antibody with the desired function. But in our ENAP, we do not consider the affinity in the antibody product as a factor in antibody generation. So using the ENAP, the molecular antibody that comprehensively binds to the epitope on the target molecule are first generated, and then the panel of the molecular antibody is created. So from this panel, the antibody with their desired functions are selected. So the ENAP technology can generate the antibodies that binds to the functionally related epitope on the target molecule. So this technology may be the best way to generate the therapeutic antibody candidates with the optimal profile for use in therapeutic application. So this is the detail of the epitope normalized antibody panel. And the first step is to design the experiment that considers the 3D structure of the target molecule and protein structure prediction and the molecular surface area and other information. And the second step is to generate the small antibody panel using a variety of the immunization method, each with a different set of setting. So by using our pair sequential assay, we obtained a sufficient number of the antibody to cover a wide epitope space. So in this case, the antibody containing the rare epitopes are obtained as required. So these are then paneled as an antibody that binds to the whole epitopes in the molecule. And this panel will be clustered and standardized to create the final inner panel. And this antibody panel consists of the minimum number of the antibodies that recognize each epitopes. So the next slide I will show the result of the functional antibody generated using our INAP technology. So the target is the CD30 that is known to the target of the antibody drug conjugate. So CD30 is the membrane protein with the molecular weight about 120 kilodalton, and CD30 is upregulated in the pathophysiology of the Hodgkin lymphoma and anaplastic large cell lymphoma. And the CD30 is also known to the drug target, as mentioned previously. That is used in the antibody drug conjugate. The name is the adcetris. And this is the final panel of the anti-CD30 enough panel. So the antibody to CD30 can be divided into the 9 epitopes, and we select the 2 antibodies to each epitopes. And as you can see, that the affinity that's shown in the large KD is not so weak. I think that this is enough to bind the CD30. And this is the image of the CD30 molecule and their epitopes. And as you can see, that these epitopes exist on the CD30 molecule uniformly. And as I mentioned previously, the CD30 is the target molecule of the antibody drug conjugate. So there are several antibodies used in the phase trial. And we selected these 5 antibodies and checked their epitopes. And that result shows that these epitopes are highly concentrated in the particular epitopes. But we find out that another epitope, so I want to note the activity of the antibody that binds to the other 6 epitopes. So to evaluate the function of the antibody, we use the reportage assay using the Ramsburg cell, and we overexpress the CD30 on this cell, and the NF-kappa B activity is analyzed as the indicator of the agonistic effect. So this graph shows that the vertical axis shows the NF-kappa B activity, and the horizontal axis shows the concentration of the antibody. So the HEFI1 is used for the phase trial and is known to the agonistic effect. And this in our system, the HEFI1 is also shows the agonistic effect because this NF-kappa B activity increase in dose-dependent manner. And we checked another epitope. So the antibody binds to the epitope number 1 and the number 7 shows the highly agonistic effect in this system. And we also evaluate the agonistic activity using L540 cell, so which naturally express the CD30. And we use the CD30 ligand as a positive control. And as you can see that this system shows the increasing doubling time by addition of the positive control. So the HIF1 is also shows the increasing doubling time, and the antibody binds to the epitope number 1 and number 7 shows the more potent and agonistic effect. So we can find that new epitopes that shows the more potent agonistic effect in the CD30 molecules. And the next, we evaluate the antagonistic activity using the same reportage assay. So in this case, we use the CD30 ligand as the stimulator, and the antibody is the inhibitor for this assay system. So the AC10 has been identified as the antagonistic antibody, but in our system, the AC10 shows the weak agonistic activity. But however, the antibody that binds to the epitope number 2 and the number 4 shows the significant biological antagonistic effect. So this new finding demonstrates that the unique property of these antibodies. And so I think that this result demonstrates that it is important for to further explore the functional properties of antibody target-specific epitopes. So in the case of CD30, our study shows that there is an excellent relationship between the antibody epitopes and the antibody activity. So our in-app technology for the discovery of epitopes with this function can also be used for other type of antibody production. So the following slide I will shows the new target for new targets to develop the potent functional antibody using the new format such as the biospecific antibody. So this slide shows the antibody format developed in recent years. So the most common form of the antibody drug is the canonical antibody shown here. And there has been increasing in the number of the new format in recent years. So for example, the antibody drug conjugate shown here is primarily in the treatment of cancer. So the warhead, that potent cytotoxic agent, caused cell death by conjugated to the antibody, which selectively binds to the cancer molecule. And so there is an increasing in the number of the biospecific antibody in recent years. So this type of antibody can bind to the two different molecules. For example, the one is the cancer molecule, and the other one is the molecule which express the immunological cell, such as the CD3. And also, there is increasing the small molecular type of antibody using the antibody fragment. So this antibody can alter their pharmacokinetics and their activity on the compared to the conventional antibody. So the biospecific antibody, as mentioned previously, is the molecule that can bind to two different molecules. However, the biospecific antibody, known as the biparatopic antibody have been a lot of attention. So biparatopic antibody differ from the normal bispecific antibody in that it can bind two different epitopes on the same antigen. So that this is a report of the example of the report that shows the different on the therapeutic efficacy when targeting HER2 by biparatopic antibody. So this also have created the biparatopic antibody in the extremely high therapeutic efficacy in the triple-negative breast cancer, TNBC, whereas the conventional antibodies are ineffective. So this result shows that the biparatopic antibody have a potential to generate highly active antibody that conventional antibodies cannot induce. So to make the biparatopic antibody, it is necessary to make the two different antibodies that binds to different epitopes. So our ENAP technology is a powerful tool for designing these biparatopic antibody because the ENAP can be divided the epitopes and it can select the appropriate epitopes to show the function. So the most important factor in controlling the function of the biparatopic antibody is the choice of the epitope to control the binding mode of antibody. So the next slide I will show the results that is the highly agonistic antibody shows the property that is inducing the cross-linking complex like this. And also, it can be shown that the antibody has the highly antagonistic effect. It shows the property of the binding molecules to the antibody in 1-to-1 ratio. So we choose the TNFR2 as one of our target. So because our group has reported that the structure of the TNFR2. So this, our colleagues shows that the TNFR2 requires the clustering of the ligands and ligands for the signal transduction. So in general, so the antibody is a bivalent molecule. So the antibody can cross-linking to each receptors in the case of the TNFR2. So the antibody against the TNFR2 can induce the weak agonistic activity when added to the cell at the high concentration. So to make the biparatopic antibody, we combine the 5 antibodies against the TNFR2. So the name of antibody is shown in red, and the structure of the TNFR2 is described in the light blue. And the epitope of these antibodies are shown in the red part. So here. So this is unique to other antibodies. And the next slide shows the result of the function of the biparatopic antibody of their function. So this is a result of the function of the biparatopic antibody. So firstly, we shows the conventional antibody that binds to the TNFR2. So this top graph shows the agonistic effect, and the bottom graph shows the antagonistic effect. So we use the same reportage assay system that express the TNFR2 on the Lamsburg cell, and NF-kappa B was evaluated as the indicator of agonistic effect. So as you can see that these 5 monoclonal antibody shows the weak agonistic activity, because I think that this is the result of the cross-linking activity to each receptors. And the one antibody and shown in the name is TR119 shows antagonistic activity, and this result shows that the antagonistic epitope is limited on the TNF receptor molecule. And then we evaluate the agonistic and antagonistic effect on the 10 biparatopic antibodies. And this is a result of the effect. And there are 6 biparatopic antibody shows the more potent agonistic effect compared to the monoclonal antibody. So this and also the antagonistic effect is shown in the 2 antibodies. And interestingly, this antibody, the name is the BP109-19-2, shows the highly agonistic antibody, but the non-agonistic antibody is shown in this antibody, and the other one shows the weak agonistic activity compared to the conventional monoclonal antibody. So I want to note the mechanism of this agonistic and antagonistic effect on the biparatopic antibody. We measure the molecular size, that is antibody-antigen complex size, by multi-angular light scattering. And the antibody shows the agonistic effect is the about the have the molecular size is the 300 kilodalton hour and more. The result shows that this type of antibody with the agonistic effect shows the complex, the multiple complex shown in here. But the antibody without the agonistic effect shows a molecular size about 180 or 190 kilodalton. This is a more smaller than this complex. And this shows that this complex is formed like this, the 1-to-1 ratio binding in this the complex. So this result demonstrates that the signaling from the receptor could be controlled by changing the binding mode of the antibodies. So this is a summary of our research, and using a technology called the ENAP, we were able to generate a set of antibody that comprehensively binds to molecules that are drug targets. And the ENAP technique is also useful for the generation of biparatopic antibody. So by creating the biparatopic antibody that can bind different epitopes individually, it is possible to induce the stronger activity that is different from the natural antibody. So we believe that our ENAP technology can make the significantly contribution to the creation of the functional antibody with such potent activity. So this is the acknowledgement, and I want to say the thanks to our groups in this institute. And also, thank you very much for your attention.
Dr. Kamada's talks open for discussion. And I have a question regarding the activation, because when you look at the pure agonist and antagonist, it must be related to the activation of your receptor, right?
Yeah.
So when because in your study, still you have to use the reporter assay to differentiate them.
Yeah.
Can you use the structural information to compare the activated receptor and resting receptors?
So we didn't activation form of the complex because this is a membrane protein. So we measure this soluble receptor to analyze their conformation by the cryo-EM. So the result is almost same of this type of complex because this is the DNF receptor 2 and this is a receptor. This is the antibody of the this against the DNF receptor 2. So this type of antibody can bind to the same direction to the TNR2. So this is the I think this is correct to the form of the antagonist effect, but we didn't we cannot analyze this type of multi complex because the cell membrane they have the cell membrane, so only this type of complex is analyzed by multi-angular light scattering.
I see.
That is all.
So, any questions?
Yeah, thank you for your wonderful talk. Yeah, you use the ENAP technology to generate antibody. So, as we know, antibody. Sometimes we always found it's against the immunodominant epitope. So, focus on very few epitope. But in your strategy, you can use your technology to generate antibody against the different epitope in the whole molecule.
Yeah.
So you use the hybridoma technology or phage display or the other technology. Yeah.
So we use the hybridoma technology to make this antibody panel. So we make the some amount of the antibody to generate the inner panel, but finally we select the good epitopes and the antibody against these epitopes. So this is this panel consists the minimum number of the antibody in each epitopes. So we use the hybridoma method to make this one.
So I have a similar question, because in your study you have to generate so many antibodies to probe the different probes. So what is the minimum numbers of the epitopes and antibody for this technology?
So in the case of CD30, the two antibodies is enough to evaluate the function. But if the more big molecule, we choose the more antibodies for each epitopes, maybe.
So it's a case by case?
Case by case, yes.
Okay. So any questions for Dr. Kamada?
Thanks for your wonderful talk. I just have questions about the TNF-alpha-2 receptor 2. I don't know if I miss it. It's due to the antibody cross-link the TNF-alpha-2 and cause the agonist function?
Sorry?
I mean, you say the antibody could be the agonist, right? So it's due to the antibody cause the TNF-alpha-2 kind of cluster or cross-link and then cause the activation of TNF-alpha-2? Or did you compare this kind of agonist with the natural TNF-alpha ligand? What's the difference?
So I think that TNF-alpha-2 can make the cluster for signaling the in the intracellular signaling. So in this case, the antibody can cluster, make the cluster in the cell membrane.
I mean, what's the difference between the natural lichen to induce the cluster? Because if just cluster, then directly can activate. So what's the difference between the natural lichen?
So I think that the bioparatopic antibody is easy to make the big cluster. So because the two different epitopes can bind to the this bioparatopic antibody. So this is the this bioparatopic can make the big cluster. So the agonicity is more potent compared to the natural antibodies.
Okay, maybe I just simply ask, did you compare the TNF-alpha binding mode and the antibody binding mode? Did you compare?
Okay, so I didn't compare the natural TNF ligands and antibodies, but I think that this bioparatopic antibody is more potent compared to the TNF ligands.
Okay, thank you.
Okay, probably I have another questions for you. I mean, the activation of the receptor, right? Yeah. For example, like a TNF-alpha, it will cause clustering, right?
Yeah, yeah.
But for some receptors, they won't cluster. They simply just, you know, have the conformational change. So, I mean, there are different kinds of agonists, or I always say antagonist or agonist effect. So with these different types of activation, do you have a technology to differentiate those different effects?
Yeah,
with clustering or conformational change?
So in this case, TNFR2 is the making cluster,
yeah.
But I think the in the case of CD30,
yeah,
it may be the conformational change in this antibody, but we didn't check the change of conformation in this CD30 and antibodies. Yeah.
Hi, nice talk. For ADC, the rate of endocytosis is maybe very important. I don't know if you checked the antibody bind to different epitope of, for example, CD30. Do they have different rate of endocytosis?
Yeah. So in the CD30, we checked the internalized antibodies. So the AC10 is known to the agonistic effect and use the antibody as the antibody drug conjugate. So this AC10 is the number one internalized activity, and the other one is not so strong. So maybe the AC10 is selected by the internalization assay to use the antibody drug conjugate.
Okay, no more question. Let's thank Dr. Kamada again. Yeah, thank you.
Thank you very much.
So our next speaker will be Dr. Lai Mingtian. He's quite famous in Taiwan. He's from Haoding, OBI, okay? And Dr. Lai got his PhD from University of Minnesota and did his postdoc at MIT. And he developed many drug candidates. According his CV, he developed 12 drug candidates in clinical trial. And one of drug candidates is in market now. And OBI is very famous in Taiwan because, you know, they kind of pioneer for drug development in Taiwan. So let's welcome Dr. Lai.
Thanks, Dr. Zhuang, for the kind introduction. And I would also like to thank the organizer for the invitation. So in my today's presentation, I will introduce a new ATC, OVI-982. It is an anti-TROP2 ATC with distinct properties. Okay. So here is the agenda for my talk today. First, I will introduce the results from characterization of a novel TROP2 antibody, 4702. So this antibody is the antibody that we used to develop the ATC OVI-982. So followed by presenting the results from characterization of this of OVI-982, and last part of my talk, I will focus on the anti-tumor activity of OVI-982 in various animal models. So first, in the characterization of novel TROP2 antibody 4702. In this section, we'd like to know what's the difference of 4702 from other anti-TROP2 antibody. So first, you may ask why we target TROP2. Because it's well documented that TROP2 is not only highly expressed, but also highly prevalent in all kinds of cancers. So TROP2 is a target that has been intensely pursued for the cancer therapeutics, especially in ATC format. So currently, there are two most advanced ATC, such as sacituzumab govitecan, abbreviated as SG, and Dato-DXd. So for the SG, it has been approved by FDA for treating triple-negative breast cancer as well as bladder cancer, and Dato-DXd, the BLD has been submitted for non-small cell lung cancer and currently under FDA review. So the antibody for SG is the sacituzumab, and the antibody for datopotamab, Dato-DXd is datopotamab. And SG have the linker of CL2A, which is not very stable because it's pH sensitive and hydrolyzable. And the Dato-DXd, the linker is GGFG. And both ATC use the topoisomerase-1 inhibitor as a payload. So the SN38 for SG and deruxtecan for Dato-DXd. So I'm going to refer SG and Dato-DXd and antibody sacituzumab and datopotamab frequently later in my talk, because we use them as benchmark for a comparison purpose. So for ATC, there are three important components to construct the ATC. They are antibody, linker, and payload. So to develop a good ATC, the antibody have to have high affinity and high specificity to the target. For linker, linker need to have good solubility to prevent potential aggregation during process, and also need to have good stability during the circulation. And for the payload, of course, you need to have a good potency, also have good hydrophilicity as well, for a reason the same as the linker. So all these three components all play a very important role in impacting the efficacy of the ATC in clinical setting. So once this ATC binds to the target, binds to the antigen on the cancer cell surface, it will then internalize into this cancer cell and then release the payload to kill the cancer cell. So the antibody that we select for OBN982 is R4702. So we first identified the epitope for the R4702. We found that R702 binds to the epitope that's distinct from the department and cetuximab. R4702 binds to the cysteine-rich domain, and the department and cetuximab bind to the cysteine-poor domain. So we then assess the binding affinity of these three antibodies to the TROP2 protein. And we, based on the residence plasmon and based on the surface plasmon residence measurement. As you can see here, the R4702 has much better binding affinity compared to cetuximab and datapolamide. It is 7-fold higher affinity than cetuximab and almost 100-fold better affinity compared to datapolamide. Because R702 has faster on rate and very slow off rate. However, interestingly though, when we assess the binding affinity of these three antibodies to the TROP2 expression cancer cell, showing here the MCF7 and also NCI N87, as you can see that the binding affinity actually quite comparable among these three antibodies. So you can see that the R4702 maybe only a little bit better, have better than the datapolamide or cetuximab. So we also have the X-ray structure for datapolamide, cetuximab, and R4702. So when we compare the X-ray structure of datapolamide and cetuximab, we found that the X-ray structure are actually very similar between the two antibodies. On the other hand, when we compare the X-ray structure of R4702 with cetuximab, we found that there's a big difference in CDR-H3 region. So you can see the pink loop for the R4702 is much smaller compared to the green loop of cetuximab. So this suggests that R4702 have larger binding space in this region. So we then monitor the internalization rate or internalization level of R702 and the other two antibody. As you can see in this result showing on the left-hand side, that R702 actually have lower internalization level and internalization rate compared to datapolamide and cetuximab. But we also use trastuzumab as the control, and you can see here that the internalization rate or internalization level actually lower in the case of trastuzumab. Excuse me. So, as you know, that trastuzumab is the HER2 antibody that is used as the ATC, which is the antibody used the FDA-approved ATC in HER2 for treatments of the breast cancer. And HER2, as you know, that actually is an excellent drug for treating breast cancer. So based on this result, it may suggest that the internalization might not play a very crucial role for the efficacy of ADC in clinical setting. So we have the antibody R702. How about the payload? What kind of payload we are going to use? We use the exatecan as a payload because it's more potent, it's a few-fold more potent than the other two payload, DXD and the SN38. More importantly, the reason we select exatecan as a payload because it is less sensitive to transporter-mediated resistance. Because a lot of time for cancers, the tumor to develop resistance to chemo because they have transporter-mediated resistance. So as shown in this table, that exatecan is a weak substrate for a transporter BCRP. It's not a substrate for a PGP. On the other hand, DSD is the substrate for BCRP and PGP. And SN38 is a substrate of BCRP, but not a substrate of PGP. In addition, exatecan also have higher cell permeability. So this suggests that it may have better bystander effect. The so-called bystander effect that when this payload used in ATC, they may have the capability of killing target negative neighboring cell. So I will show that later in my talk. So we have the antibody, we have the payload. How about the linker? So we designed the hydrophilic linker for OBI992. So OBI992 have a novel anti-TROP2 antibody with distinct epitope, with distinct structure. We use the hydrophilic cathepsin B cleavable linker, and we have a highly potent payload, which is less sensitive to transporter-mediated resistance. And we also optimize the conjugation process to reach maximum percentage of drug-antibody ratio of 4. So we have this ATC in hand, then we carry out the characterization of this ATC OBI992. So first, we evaluate the cytotoxicity of OBI992 and DXd in different cancer types with different level of TROP2 expression. As shown in the table A, you can see the IC50 of the OBI992 and DXd are quite comparable when tested in all these different cancer types. And we also found that the IC50 actually have close correlation with the TROP2 expression level shown on panel B. So we also want to know that this ADC have off-target toxicity because this often contribute to a narrow therapeutic window. So first, we obtain the leukocyte from 2 donors and isolate the lymphocyte, monocyte, and granulocyte from the leukocyte and assess the association of OBI992 and DXd. We can see that DXd has much higher association with monocyte. And furthermore, we assess the toxicity of OBI992 and DXd in the TSP1 cell. The TSP1 cell is the Fc gamma receptor bearing cell, and it has been documented that ADC that associate with the
Fc gamma receptor cell would cause toxicity. So we assessed the cytotoxicity of OVA992 and DETO-DSD. As you can see from this, from figure B, that DETO-DSD have 6-fold more potent in killing the TSP1 cell. In addition, we also assessed the cytotoxicity of OVA992 and DETO-DSD in differentiating neutrophil. Again, in this study, that DETO-DSD have 30 more potent than OVA992 in killing differentiated neutrophil. So this result suggests that OVA992 have lower potential to cause off-target toxicity compared to DETO-DSD. So in next few slides, we actually want to evaluate how stable of OVA992 in vitro and in vivo. So as shown in this slide, on the left-hand side is the free payload that detected when the serum incubated with OVA992, and we got a serum from different species. And right-hand side is the percentage of free payload detected in the serum when in the study with DETO-DSD. So if we pay attention to the purple line, which is the human serum, so the percentage of free payload in the human serum is lower than 2% in the study with OVA992. However, it's almost reached up to 8% in the study with DETO-DSD. So this suggests that OVA992 is stable in the serum, but we are asking the question, will this result, in vitro result, can be translated into in vivo? So we conducted in vivo pharmacokinetic study in vivo in rat. So as you can see in the left-hand side, that the blue color is the total antibody. The red color is ADC. You can see these two lines are very close to each other, suggesting that the ADC is quite stable. On the other hand, when you look at the right-hand side, that study with the DETO-DSD, those two lines actually separate a little bit far away, suggesting that ADC is not that stable. So this is supported by the level of the free payload detected in the circulation. As shown in, excuse me, as shown in the left-hand side, that the free payload is much lower compared to the free payload detected in the study with DETO-DSD, and you can see that at the early stage of the incubation, the payload actually released to very high level. So in terms of the Cmax detected in the study with DETO-DSD is 4-fold higher than the Cmax of the free payload in the study with OVA992. So here is a summary of this pharmacokinetic study. You can see that the exposure of OVA992 is 2-fold as the exposure of the DETO-DSD. On the other hand, the free payload of OVA992 is 50% of the free payload in the study with DETO-DSD. So this suggests that indeed OVA992 is more stable, either in vitro or in vivo. We continue to ask the question that this ATC is more stable in the circulation without actually reflect in the tumor situation. For the mice that bearing the tumor, where the ATC have, with the tumor, have higher concentration of OVA992 or also have higher concentration of free payload from OVA992. So to answer this question, yep, we carry out a study in the animal-bearing tumor. So this is a study that in NCI 1975 is lung cancer CDX model. So in this case, on the upper panel is the concentration of ATC, total antibody, free payload in the serum. The lower panel is the ATC, total antibody, and free payload in tumor. So as you can see, the blue line and red line is also much closer to each other in the case of OVA992 compared to those two lines in the case of the DECTO-DSD. More importantly, let's focus on the concentration of ATC, total antibody, and free payload in the tumor. So you can see that based on the time-dependent monitoring of the blue line, red line, and purple line, that representing to the total antibody, ATC, and free payload. So it's clearly that the exposure of the antibody, ATC, and free payload is much higher in the study with OVA992. So based on the calculation, that is about the twofold higher in the study with the OVA992 than the study with the DECTO-DSD. So we further monitor the free payload accumulation in the tumor as well as the other organs. And shown here that you can see that the free payload accumulation in tumor is much higher in the case of OVA992 compared to the case in the DECTO-DSD. So based on the calculation, the accumulation of the free payload in the tumor is twice higher in the study with OVA992 compared to the DECTO-DSD. So based on the study, the in vitro characterization of the OVA992, we found that the OVA992 have low potential of causing toxicity and also have better PK profile, and more important, has higher accumulation of the ATC and free payload in the tumor. So last part of my talk, I will focus on the anti-tumor activity of OVA992 in various animal models. So first is the cell-derived xenograft model. It's the triple negative breast cancer model. You can see that A, B, C panel represent different doses of the ADC. And for SG, we dose 12.5 mpk twice a week, which is consistent to the study that conducted by Gilead. So as you can see that at a dose of 10 or 3 mpk, the anti-tumor activity are quite comparable to each other. In the panel C, when OVA992 dose at 1 mpk, the anti-tumor activity actually better than DECTO-DSD in this study. So we also carry out the anti-tumor activity in another model, that BXPC3 pancreatic cancer model. In this case, they all dose the same dose, 10 or 3 mpk, and the dosing schedule is once a week. From this study, we can see that both OVA902 and DECTO-DSD have better anti-tumor activity compared to the green line SG. So in the NCIH 1975 non-small cell lung cancer model, the OVA902 actually have clear advantage in the anti-tumor activity at 10 or 3 mpk compared to DECTO-DSD and SG. So we conduct this anti-tumor activity in in 8 CDX models. And here is a summary of the study, the result from a study. And you can see that at 10 mpk or 3 mpk OVA902, OVA902 have remarkable anti-tumor activity, except that in this NCIH 1975 C797 CDX model is only reached 60% of anti-tumor growth inhibition at 3 mpk. So in addition to our CDX model, we also carry out the anti-tumor activities study in a patient tissue-derived xenograft model, PDX model. So here showing 4 examples of this study. The panel A in the lung cancer model in this study showing that at the high dose of OVA902 have a better anti-tumor activity compared to DECTO-DSD. In other lung cancer PDX model, it actually both OVA902 and DECTO-DSD have comparable anti-tumor activity. So in another model, panel C, the gastric cancer model, that at low dose 3 mpk of the OVA902 have better anti-tumor activity compared to DECTO-DSD. For the breast cancer model, panel C, actually both OVA902 and DECTO-DSD have comparable anti-tumor activity. So we conduct this anti-tumor activity study in 13 PDX models, and here is a summary of the results. So again, OVA902 show remarkable anti-tumor activity in PDX model, except that one of the pancreatic cancer model that only achieve 65% of tumor inhibition at 10 mpk. So this is the anti-tumor activity study. Then we also we want to know that OVA902 or DECTO-DSD is capable of suppressing the tumor growth in large tumor model. And with the mice that treated with respective ATC, what kind of the survival rate? So in this model, we actually intentionally that the tumor grows up to 450 or 500 millimeters cubic, and then treat the mice with the 902 or DECTO-DSD. In both doses, the OVA902 have better anti-tumor activity. When we monitor the survival rate of the mice, that for both doses, 10 or 3 mpk, the mice treated with OVA902 have longer survival rate compared to DECTO-DSD. So we then want to demonstrate the advantage that we use exatecan, not SN38, not deruxtecan, because as mentioned earlier, exatecan is a weak substrate of the BCRP, is not a substrate of PGP. We're going to demonstrate that it has its advantage in the anti-tumor activity. So to test this hypothesis, we use the animal model SCC827, and you can see that all three ADCs showing the comparable anti-tumor activity in parental cell. When they overexpress PGP in the cell, you can see that OBI992 have better anti-tumor activity compared to DECTOR-DSD because the payload that used in DECTOR-DSD is a substrate of PGP. Then we test that when we overexpress BCRP in the cell, what will happen? Not surprisingly, that in this case, OBI992 have better anti-tumor activity than SG and DECTOR-DSD because the payload they use, they all are substrate of BCRP. So this result suggests that indeed OBI992 would be less sensitive to transporter-mediated resistance. So another study which is very critical for ADC is the bystander effect. So given the heterogeneity of the tumor, it's very important for ADC to have bystander effect. So so-called bystander effect is that your ADC is capable of killing target-negative neighboring cells. So to prove that OBI992 have strong bystander effect, we use the TROP2 negative ES2 cell. So you can see on the left-hand side that both 992 and DECTOR-DSD did not have anti-tumor activity against the ES2 tumor because they are target negative. They don't have TROP2 to target on. However, on the right-hand side, when you mix ES2 cell with TROP2 positive BXPC3 cell, we show that significant anti-tumor activity for the DECTOR-DSD and 992, because this suggests that once the 992 or DECTOR-DSD kill the BXPC3, the payload actually can dig out to kill the neighboring target-negative cells. And when we look at closely at the day 15, the OBI992 show a little bit better anti-tumor activity than DECTOR-DSD. So we believe that if we carry out the study longer, we may show better bystander effect of 992 compared to DECTOR-DSD. But at this point, it's not statistically significant. So next, we'd like to see what kind of combination we can use in future clinical trial. So one of the hypotheses is that the topoisomerase-1 inhibitor would cause double-strand DNA double-strand break, which would trigger the DNA repairing mechanism, and PAR1 protein is one of them. So in theory, if you combine the ADC containing topoisomerase-1 inhibitor as a payload, could show synergistic effect in anti-tumor activity if we combine with PAR1 inhibitor. So indeed, that's the case we observed in this study. So the PAR1 inhibitor we use in this study is talazoparib. And you can see that we don't see much of the anti-tumor activity at the dose that we use for OBI992 and DECTOR-DSD, the talazoparib individually. But when we combine them together, we show significant anti-tumor activity. So suggests that there's strong synergistic effect in the anti-tumor activity. Furthermore, we also want to see if we can actually combine with immunocheckpoint inhibitor, because we in-house we demonstrate that OVN-982 able to induce immunogenic cell death, which suggests that it can activate once the 992 killed tumor cell can activate immune response, which is suitable for combination with the immunocheckpoint inhibitor. So we test this hypothesis by combining the OVN-982 with anti-PD-1 antibody, and we do show the synergistic effect in anti-tumor activity combined with the anti-PD-1 antibody. So this is an anti-tumor activity study, and in the end, we carry out the GLP tox study in cynomolgus monkey. And the dosing regimen, 6-week dosing, 6-week recovery, and once every 3 weeks for 3 times. And the dose are 0, 20, 40, 60 mpk. And the highest non-severe toxicity dose was determined to be greater or equal 60 mpk. Because the 60 mpk is higher dose, we win. So when we compare to the HNSTD of Dettol 10 mpk and SG 50 mpk, we may have a broader therapeutic window. The major finding is scaly skin, pigmentation. There's minor impact on hematology, but they all are reversible during the recovering period. So we actually submit the IND application last year, and we received the safe to proceed from FDA early this year. So right now, OVN-982 is currently in the phase 1/2 study, and we also present 3 posters in AACR this April this year. So this is the summary of my presentation, that OVN-982 have the potential to be best-in-class anti-TROP2 ADC. So I'm not going through each bullet because I just present those data. So I will conclude my presentation here, and I will be happy to answer any question you have.
So Dr. Light's talk is open for discussion. Congratulations. I mean, you in your, I would say, study, when you use 2 drugs, you can increase the anti-tumor effect. Is that possible? You can make like a 2 payload ADC.
Yeah, that's actually in consideration. We are working on that.
Oh, okay.
Yeah.
Thank you for that very comprehensive study. So my question for you is, so your ADC is different from the comparators in terms of the antibody itself, the linker, and also the payload. Your antibody is so 100 for higher affinity than your comparators antibody. Do you think it is the better efficacy in animal studies? Is it contribute to your higher affinity antibody, or your linker, or your payload? And can you break down so why your antibody is better than the comparators?
Yeah, as I mentioned, that for the efficacy of ADC, really multifactorial. We cannot actually single out just or antibody alone, or the payload or linker, actually they all play a crucial role in impacting the anti-tumor activity for ADC. Like, for example, that if your linker is stable, that's contribute quite a bit, because when we compare to SG, they use the CL2A, which is very unstable. In our early animal study, it's really not showing a good anti-tumor activity. We also do the study by replacing the tetroDSD with our linker, and we actually can show better anti-tumor activity. So it's multi-factor. We cannot just exclude that because this single factor contribute to all the activity.
As a follow-up, that is in the ADC field, so people believe that you do need a very high affinity antibody, because that's potentially can have a potential side effect. Of course, in your case, you showed better efficacy, so that's a good example. But so what's the reason you initially decide to drive the antibody to such a higher affinity?
It's actually so the high affinity is one thing, but I have to say that not necessary, because high affinity is pros and cons. Because although the tumor cell express high level of TROP2, but don't forget, our normal cell also have TROP2. So when we select the antibody, we need to be careful. You don't want to select extreme high affinity, because you actually ask for more off-target toxicity.
High affinity, right?
Yes.
Yeah, so that's why I ask you, so when you design your experiment at the beginning, you must have a panel of antibodies with different affinities. You pick the one with a very high affinity. And so the question is precisely, the potential for off-target activity.
Yeah.
But in your case, you're lucky the antibody works well. You animal models, you face one clinical trial.
Okay. Yeah, that's a good point. And actually, when we test the cytotoxicity in the tumor cell expression, the TROP2 target, we see the comparable cytotoxicity. So that's actually that's a relief to us, because again, we don't want the really high affinity. So yeah. So in the cell-based assay, that is a relief to us. Yeah.
Thanks for an excellent talk. I just like to ask about the logic when you combine this drug with the anti-PD-1 antibody. I don't know. I assume pembrolizumab or yeah. Anyway, that type of antibody. Then how do you determine what kind of relative ratio to find the best synergistic effect?
Well, so when we designed the study, we actually have to make sure that the initial combination dose didn't really see the good anti-tumor activity individually. We have to make sure of that. And then when we combine together, we hope to see significant anti-tumor activity. So we test out early on, let's make sure that in this dose, we don't see significant anti-tumor activity, and then we combine together to see if we actually have a good synergy. Yeah.
In your PK study, if I understand the data correctly, it looks like the free drug concentration is much, much lower than the ADC. Does that mean that like less than 1% of the ADC is being activated? And if so, is there some strategy to increase that? It seems like that could make a huge impact on the therapy. I don't know if you understand my question.
So the free drug in circulation
in the tumor.
In the tumor, yeah. So
it's like much lower than the ADC, right? It's like 10 nanogram per gram. But the ADC is like about 1,000 times higher.
Yeah.
So does that mean that only less than 1% of the ADC is being activated? Is that what that means?
Actually, based on the calculation that for the percentage of the payload really get into a tumor is less than 1%. That's
okay. So that seems like there's some room for big improvement if that can be overcome.
Exactly. That if we can increase the intake of the payload or ADC in the tumor, that would be great. That would actually we are looking for. Yeah.
I think time's up. Let's have a break. Thank you.
Okay.
Thanks again for two wonderful talks and the moderator, Dr. Zhang. It's about time to have a coffee break. Please come back to the third floor, enjoy your coffee, and come back on time. Thank you.
Right?小背景先关掉是吧？没有，我是要用电脑。不会啊，超好，超强。 就是我们是有接的这个 integration 的这个，然后它的有一个镭射，就是看一下这样。那如果这个觉得不够强的话，我们还要准备是更强的镭射。对，可是这个不能翻页，它不能 next page， 所以就是看你这样子，看你觉得怎样比较好。操作看看。对对。我没有特别担心，这还好。辛苦了。好的，那我们等一下会，因为这个休息比较长，到3点半。那如果你有时间可以到这边去休息，知道我们在的时候就在3楼的那个coffee break，你知道在那边，就是您从也可以，或者是您从这边出去坐个电梯上去，然后我们有。 在前面的这里。
Ladies and gentlemen, the conference will resume in 3 minutes. Please have your seat. Thank you. Ladies and gentlemen, Ladies and gentlemen, it's about to begin. Please have a seat. Thank you. It's our great honor to have our next moderator, Dr. Zhang Dachi. She is the co-founder and CSO of General Biotech Company. Please welcome her.
Today, the last section, we are going to introduce to you new modalities of treatment, especially gene therapy and Prozac. Okay, so first speaker would be Dr. and Professor Wu Lianghu, and his educational track record are all established in National Taiwan University, the very prestigious one in Taiwan, and also he has a pretty impressive experience in the United States at Johns Hopkins University and Mayo Clinic Center. So today he is going to share with us the very first gene therapy successfully developed under his hands in person. So we please give him a big applause for the first-hand story. Thank you.
So thank you for inviting me to this meeting. So I will change the word from antigen, antibody to gene. But actually, I previously got some connection with the antibody production. So anyway, I feel very warm to sit here and talk to you. So this is always the first slide of my teaching slides. So I'm teaching medical students in school for like 30 years. Yeah. So when we introduce something new, we always give a comparison. This is something old. So this is the oldest the medicine in Chinese. Yeah. So this is Shenlong. Shenlong just taste for many herb drugs to find one that correctly cure the disease. But even though you think this is old, actually we are still doing that. We do small molecular screening. So we screen for hundreds of thousands of small molecules, try to match one of them to cure a phenotype. We do high throughput screening. So it's the same notion. But certainly some of you also screen for monoclonal antibody. This will screen a lot, yeah. But this is in contrast to this. It's not targeted. It's want to find by random selection. But I think the point of this meeting is the antibody, because antibody got this very strong specificity. So antibody could be used for many purposes. So I just mentioned, for example, is CAR T. So is it still antibody? You just try to transform the antibody to a T cell, then totally you change the treatment. So for us, we do gene therapy, we think of CAR T is a gene therapy. So it's a gene-modified cell therapy. Okay, it's not a pure therapy, yeah. But this is many things in the antibody. But there is another molecule in the world. It is very specific. It's our DNA. It's a double helix. So if you denature the double helix, it will anneal back exactly. So even though you use a very short oligonucleotide, it's only 20 base pair. So you can use that as a drug. That drug will find the target in our cell. So even 20 base pair can have a kind of good specificity to target. If you think that 20 is not good enough, because 20 is for any base, we have 4 possibility. So 20 is 4 times 20 is potential. You can use 2 oligonucleotides, and each end adhere with half of the nucleus. Then you have a 40 base pair specificity. It's almost unique in our genome. Yeah. Okay. So then, how we play with the gene therapy? So in this slide, I want to expand this field a little bit. So either you target the gene, or you use the gene, use DNA, use RNA as a drug to treat the patient. I call all of them as gene therapy in this lecture. I just introduced from this slide a few of the technology that can be involved in gene therapy to you. For example, the most we use is the gene transfer. We can use a viral vector, and the list here is adeno-associated vector. We call this AAV. It's a very small virus. But certainly, maybe you are more familiar with adenovirus because we use that to produce a vaccine. The AD clonal vaccine. For others vector, we can use the lenti-vector. For example, this is for CAR T. You need to use a lenti-vector. But certainly, we can use a non-viral vector, like a liposome or nanoparticle, to deliver a nucleic acid. We call the nucleic acid, it could be DNA or RNA. I think nanoparticle LNP is very famous currently. A lot of people, including people in Taiwan, try to devote to find a new nanoparticle. But unfortunately, until now, they are except for vaccine, there hasn't been any major success of LNP in medicine. We mentioned a little bit of gene editing because we say that the specificity of the DNA, the best example is to use CRISPR-Cas9. For this technology, we use RNA to recognize your target is 20 or 18 base pair, and then that molecule will bring an enzyme Cas9 into the specific location to cut the DNA. And then from that, we have base editing, primer editing. Again, I just told you that with very small RNA, you can change a lot of things. With the 20 base pair RNA, you can suppress a gene, activating a gene. You can change splicing. A lot of diseases, they are concerned with splicing. So they are very convenient. And sometimes you can say that this nucleotide is like a small molecule. It's really not like a big molecule. Okay, so I then convert the technology in this slide and try to demonstrate their role in our genome. So this is the DNA, so we have exon, intron, and then we have a translation, heteronuclease RNA, splicing into message RNA, the polypeptide then folding. So there are a lot of possibility that we can change this process rather than just targeting the end process. The most successful things we currently done is just replacement. It's not really very fancy, but we can do that with kind of confidence. Yeah. So then with replacement, we use a different kind of virus, nanoparticle, but certainly we can that. And then we can either bring the most conveniently is bring down the expression of some gene. It's easy that we use this small RNA. You can use another bigger or stronger technology like CRISPR-Cas9. But as I say, it's easier to bring down rather than activate. Yeah. But you say that this not fulfill the disease model of my disease, the disease that I interested. But there are many examples that people can find during the pathway that rather than activating the gene that usually others target, we can find other place that we do a suppression. Then, as a consequences that we achieve the treatment effect. For example, if you lost a suppressor, then everything got activated, then you can suppress one of the downstream gene. So there's many of these examples that we can talk about. But certainly with the newest base editing, now we can modify sequence. I've said that with a very small RNA, we can change the splicing. And then we can just make any DNA, any RNA you want that we synthesize. We can do combination. We can change. We can evolution of a protein. I change the protein and make it fit your purpose. So there are a lot of things that if you're starting from the gene, it's really change your world. Yeah. So then we talk about my history. Yeah. So when I entered the field of genetic disease, there was not many treatment. Yeah. So the biggest thing that changed the field of genetics is the enzyme replacement therapy. So for many diseases, there are genetic disease, the gene, loss of a gene, loss of protein, and then we replace that protein because the enzyme. Most likely, the protein is the enzyme that catalyzes metabolic pathway. So you call this enzyme replacement therapy. So this is the story from the Pompe's disease. This is Professor Y.T. Chen. He discovered in Duke University, and that really changed the life of a baby. So that the baby with a Pompe disease, infantile onset type, usually they will have a very big heart at 3 months of age and die before 1 year. And with this drug, and then patient can survive. Yeah. So we are very fortunate here. We also join the clinical trial and also conduct a number of the post-marketing researches of this disease. But then we have a problem. So this protein, they don't get into the brain. So because of this blood-brain barrier, so these are many things that protect the brain from the molecule entering from bloodstream to the brain. So then we really got trouble with some of the diseases. For example, this is a mucopolysaccharidosis. We call this a 黏多糖. Okay. So for example, this MPS type 2. So this is the most common type of MPS in Taiwan, in the Asia. And there's one or two companies, they develop ERT. We are very happy to treat the patient. But the fact is that 2/3 of those patients, they have mental retardation. But actually, I really don't feel comfortable that, oh, we treat the patient, they become more mobile, they survive longer, but we really don't treat the brain. But there are many other examples that we really don't want to treat the brain. Many of the lysosomal storage diseases, they affect the brain, but certainly there are other brain diseases. That's a neurotransmitter, that's the seizure, or other metabolic disease, iron accumulation. So those are the diseases that many of them don't have a treatment. So when you are in pharmacy, if you try to count the disease without treatment, I think the most number is in the brain, is the neurological diseases. And then with ERT, we are very happy, a lot of product, a lot of meeting, but we still don't treat the brain. So I think everyone wants to think about how to pass through this. We call this blood-brain barrier, the tight junction link the endothelial cell that prevents the leakage of any of the molecule. So sometimes it's a lipid, they can just pass through it. A few things they get into the brain through receptor-mediated mechanisms. So you can try everything you want to do. So if you are very violent, you can use ultrasound to break it and to let your molecule get into the brain. Okay, so then we go back to the enzyme replacement. So how to let the enzyme, MPS 2 enzyme, MPS 1 enzyme, to push those enzymes into the brain. But certainly we can use the receptor modified method. We can modify the enzyme. But certainly we can use cell therapy. Yeah, because we know that when we do bone marrow transplantation, hematopoietic stem cell transplantation, the macrophage will migrate into the brain, into microglia, then the microglia can do something good in our brain. We will talk about it a little bit later, but certainly now we got the virus. Yeah, so I will show you that some of the virus, they will get in the brain. But certainly we have encephalitis, for example. Yeah, so the virus, some of the virus will get into the brain. So the virus, then our drug is a virus. It's a live virus. It's not a killed virus. It's not a vaccine that will kill the virus. No, we need the live virus, and they will infect your cell. Will that be dangerous? No, because of the advancing technology, now it's not dangerous. For example, if you want to make an AAV vector, so we don't use adenovirus. So we only use 3 plasmids and sometimes 2 plasmids, and we transfer the plasmid into a cell line. The most likely is this AAV293 cell. You can also use the insect cell. And then this gene, well, after transfection, the cell will produce a virus. The virus is a live virus. So when I was young, I always fancy about the virus because it's like a life between the life and chemistry. So this is very safe because we don't touch any of the virus. And other than that,
So what we want to put in is the expression cassette into the virus. It certainly will include the promoter and the one-change gene and some others. But you see here is a adenovirus, so we replace a few of the viral genome with this expression cassette, and at the same time it will inactivate the virus. It will invade the genome. If you see this is adeno-associated virus, AAV, you see the IT over here, but actually there's no viral gene at all. So all viral genome has been replaced. So then you can believe that it must be safe, for example. I think it's too slow, yeah. Okay, so after virus get into our cell, either integration or non-integration, and we can have a different method to deliver the virus. We can use a systemic injection, direct injection, and we can also use ex vivo gene transfer. This is like a CAR T. You separate the T cell, get into the virus. We call this ex vivo. Okay, we see a few of the example. So this is very typical, like a CAR T. We use lentiviral vector, but we modify the hematopoietic stem cell, and then we put the cell back into the body. Yeah. So recently there is one disease, certainly you don't know the disease. This is one of the neurodegenerative disease, and they have been approved by this lenti-vector-based gene therapy or bone marrow transplantation for easy to remember. So this method has been approved in several of the drugs. Yeah. It's just show you that so after this autologous bone marrow transplantation with a modified stem cell, a patient can have a very persistent expression of the enzyme they don't have, both in the blood and in the central nervous system. Okay, so AAV is my beloved, and so then what will happen if we infuse the AAV into our bloodstream? It's an IV injection. I will tell you that most of the virus will be trapped in the liver. It's like the lipid, like IMP liposome, they just trapped in the liver. Essentially, we don't know why. But this is something good because liver, we need the liver to do a lot of things. For example, you can see that after we put a virus that can express factor VIII, and then after IV infusion, we have the factor VIII expressed in the liver. So then this has been transferred to a few successful medicine for hemophilia. But certainly this is a lot of failures, many failures, struggling, but then they produce this excellent, wonderful picture that it is after gene therapy, and we have the factor IX rise to like 30% of normal. We know that if we have 10% of the coagulating factor, then we don't have bleeding. So most of them have therapeutic level, and it's continued to 5 years. So I think it's very reasonable for FDA to approve such a drug. So now for hemophilia A, hemophilia B, we both have medicine. This is a little bit complicated, but how about we're talking about the brains, not the liver? Yeah, but once we have the factory in the liver, then we can make the liver to produce something else for the brain, and we can make that molecule brain penetrating, as I said. So it is a lot of potential for this liver target therapy. Now we return to the brain. So even though that we say that most of the AAV infused will be trapped in the liver, but some will get into the brain for some serotype of AAV. The exact mechanism, actually, we don't know. They just penetrate the brain. And so
It's very nicely demonstrated that it's for SMA. You know that this is first very prominent the ER gene therapy in the field. And so it's a mouse model that after you infuse the vector into the blood, and then you have your motor neuron get infected. So you do nothing, just put in the virus. The virus go to the motor neuron. So this is kind of very good targeting. It's because of virus, it's not because of us. Yeah. So now we have this gene therapy for spinal muscular atrophy. But certainly we have other therapy, like small molecule and it's oligonucleotide that change the splicing. So they are also approved drug for spinal muscular atrophy. So for this disease, very, very important disease, now we have like 3 or 4 drugs available in the field. We have gene therapy, we have this CSF injection of oligonucleotide, and we also have an oral, yeah. So I'm not discouraging small molecular analysis, but certainly if you see this picture, you will say that this is the best. The business is oral, yeah, it's a really good drug, yeah. And now people are trying to produce more serotype of AAV. So maybe I have 10 minutes that are talking about my story. So my story is certainly it's a rare disease. And the enzyme is over here. And that enzyme is responsible for production of dopamine. I think every one of you know dopamine because this is the molecule that being deficient in Parkinson's disease. So in Parkinson's disease, it's because of degeneration, it's death of the dopaminergic neuron in our brainstem. Yeah. So the patient don't have the neuron, they don't have enough dopamine. But what will happen if it's a genetic defect that the patient, the baby, don't have dopamine? So this is the disease I'm facing. So those patients, if they are in the severe type, so after birth, they don't have any motor development. So they don't have head control. They cannot sit up. They cannot stand up. It is really difficult. They have all the neurological sign, and they have autonomic system problem. They have many of the problem. So at that time, there's no treatment for the disease. And we can see that this is growth curve. For baby, the most important is growth. It's no growth, it's no life. And this is normal curve over here, and you see that the patient stopped to growth after 1 or 2 years of age. So this is very, very bad disease. But certainly if you put the patient in gastrostomy, for example, other things that they will survive. And this is a motor scale, this is normal. With age, they gain motor function, and none of our patients, the severe type, will have any motor function like just straight their heads. Yeah. But the story is starting that from the study of Parkinson's disease. So the common disease always checked the most intense study. Because in Parkinson's disease, the best drug is levodopa. But after a while, after most of your DA neuron died, then you don't have this enzyme ATC. When you eat levodopa, then it cannot be converted into dopamine. You cannot eat dopamine because dopamine don't get into BBB. So wow. So there are some researchers that, then we return this ADC enzyme to the brain, and then patient can be resume the responsiveness to levodopa. I think the story is correct. But the problem is that actually we don't have the etiology of the disease. But certainly after gene therapy, patient can return sensitivity to levodopa, but after a while, the cell still died. After all cells died, then patient still cannot move. Yeah, so till now, for Parkinson's disease, there's still no successful treatment. But this treatment will be perfect for my disease, for congenital deficiency of this enzyme, because the cell, the neuron, will not die in the disease. They just don't have the function. So if we return this protein to the place that the dopamine being produced, is our brain, is our, we call this striatum, 脑核, yeah. So then the dopamine can be converted to dopamine in the patient's brain. And the patient have a lot of very high level of dopamine, so we don't need to give them dopamine. So then they certainly will have benefit from this treatment. So actually, when we hear the story, we say that it must be a success, so it must be useful. Yeah. So we just inject the virus into our brain. Okay. So this is from previous monkey study. I just show you that, but certainly we use injection, this targeting. Yeah, so it's much easier rather than a systemic injection, because 99% of virus will be eaten by liver. But after we infuse the virus into our brain, only the neuron being affected, not the glial cell. So this is another example of targeting that directed by the virus itself. So virus is so lovely, yeah. But certainly we can increase the specificity by other methods. We can change the promoter. We can even put in small RNA, miRNA into our construct to inhibit the expression of the gene in some kind of cell to increase the specificity of the treatment. Then this is in our patient. You can see that this is before treatment. It's after treatment, we use the radioisotope PET, and we can see the expression of the drug, the virus that we inject. It's very clear. But it's the best way just to watch the patient. So because of the meeting, I don't show you video. Yeah, but what happens is that when we inject the virus, we wait for 4 weeks, then patients start to move. So at that moment, we know that it's responsive. But how good will they then will follow the patient? But it is because the virus soup is like water. It's only like less than 1 meal. So sometimes it's hard to believe that it's this very strong, potent drug until you see the movement of the patient. Yeah. But it's a very long story. You know that we published in Science Translational Medicine in 2012. It's our first paper. But in that time, actually, there have been checked some attention to the field. So this is a very long story. So at that time, in Taiwan, nobody knows gene therapy. But actually, gene therapy is not a medicine at that time. Gene therapy being classified into 医材, it's a medical technology. This is 归医事处管, 不是归 FDA 管. So when we go to 医事处, it's like a 鸡同鸭讲, so this is just impossible, yeah. But we have a patient, and then we have our belief it must be a successful. So then we try to collect money, try to make some virus, and to do the treatment in our hospital. We call this a compassionate use, like things that doctor can do something to save the life of the patient. And after that, I published the science paper. And with that data, then we really get a real scientific funds. So yeah, thanks for NRPV. They give us quite a good support that we can conduct our phase 1-2 trial. And with that phase 1-2 trial, published paper in Lancet.
And that could be a success, but then we run out of money. Because NRPV disappeared, there's no national project can support. But fortunately, we present the result in a national meeting, and then we have company interested in our product, and then we license it to the company. And that company, small new star company, then being acquired by PTC Therapeutics. And then PTC support us to treat further patients. And then we collect 30 patients. So we put them together and publish one paper, and with that data, we pass through the Europe the submission. So this is a very important figure with many patients. You can see that this is the motor scale patient, AAV patient. After treatment, they have increase in their score, that they improve. And then we see that in this group, they have a steeper line. This means that they improve faster and better. We have this group that being not so good. But what's the difference? It's the age at the treatment. So then we know that earlier treatment will be better. So this is very strong. So everything, the P is less than 0.001 like that. So then we publish this in Molecular Therapy. We get the cover. And then we also do other publications that it is kind of see the brain tract. So it means that after treatment, the brain really starts to function. Yeah. And so last year we get approved from EMA. So this kind of historical success. At that time, we don't really success in FDA because FDA asking for additional data inside the US. So PTC spent some effort to conduct another trial in the United States. This is our celebration of the approval from EMA. I say that this is the success from the patient. It's the patient's success, yeah, because they have the treatment. So just last month, after PTC performed another trial in the United States, it's only the short trial, so they want to prove safety and also the catheter that is a registered catheter. Then we jump this, and we get into this final slide. So this is a talk by Francis Collins. It's last year in the ASGCT paper. So they first congratulate for everyone. It's a gene therapy field that you treat a lot of disease. They say maybe 500 diseases that now can be treated. But there are 5,000 genetic diseases. So we still have 4,500 diseases that don't have a treatment. Yeah, so I think we need all your efforts to help those patients, and certainly for myself, I'm still trying to working and to develop more gene therapy. So thank you very much.
Thanks, Dr. Yeah, thanks for an excellent presentation. I'm sure the audience may have some questions to ask about this gene therapy. Well, compared to the usually we say precision medicine. Usually we talk about target therapy, things like that. So if you do have a choice, do you think gene therapy is relatively easier and at a reasonable cost for the patients? I mean, for the overall production compared to other, say, monoclonal antibody thing?
I will say yes. If you look at the development of the gene therapy drugs, you can say the success rate. We say small molecular development, the success rate is less than 1%.
But I think for gene therapy, it's like a 20-30% they can success. But success rate high is high, but it's not mean easier. It's still cost a lot of money. So the biggest problem that I have 5,000 diseases potentially. Many of them can be treated, but it's very expensive. The development is very expensive, and then once it's been approved, the drug is very expensive, and reimbursement is a problem. So I think there are still a long way to go. Yeah.
Yes. Yeah. Congratulations for your success here. So my question to you is, how often you have to inject the virus in the brain? So what's the dosing frequency?
You mean the
treated patient? You give the
yes, after
injection.
Yes.
And you have to give one dose, or you have to give continuously?
Yeah, so I think that the merit of gene therapy, and also we want to show up is a single treatment. Okay. So for all gene therapy, it should be a single treatment. Otherwise, it cannot justify its price. Yeah, but for this disease, but for AAV. For AAV, because it's not integrating, because of time, I didn't really explain for the detail, but this is not integrating, so people always ask how long it can sustain. But for my disease, I transduce the neuron. As long as the cell don't divide, they don't die, supposedly the episome will be there. But the problem is the liver gene therapy for the hemophilia. And we know the hepatocyte is still turnover. Maybe they say that for hemophilia, maybe 5 years, 6 years, they may lose their effect. We don't know. We still need to watch. Yeah.
Okay, the last question.
So thanks for the wonderful talk. So as you said, there are so many genetic diseases or rare diseases. And how did you pick up this one, ADAC, as your choice? What kind of decision procedure? Is it personal, or you did some kind of statistical analysis, or somehow you decide to spend so much time on this disease?
So I have many patients, many kind of disease. I always want to find the treatment for those patients. But this is a very special story. I need to spend another 2 minutes. Is that one day a father of a patient, he showed up in my clinic, and they show me a news. The news is that the gene therapy for Parkinson's disease was conducting in Japan. So then the father asked, the father is a pharmacist. They can see the keywords is AADC. So they use AADC. My child don't have AADC. Well, that be a treatment for his daughter. So this is the reason I chose the disease. I say, oh, this should be wonderful. Then we wrote to the PI in Japan, and he returned to us the same day. So then we start, yeah.
Thank you so much.
Yeah, so thanks for sharing this beautiful story with a big success in reality. Thank you very, very much. Okay, now, and now for our ending section, we are so glad to invite Chief Scientific Officer Chen Shuzhen. She actually has a strong background in pharmacy and biochemistry in Taiwan and also in the States. And then she's been a co-founder of ACGT, 行动基因, and then successfully helped from discovery to product development, to IPO and to national insurance reimbursement. Yeah. So and then she shifted her gear back to drug development. So now she's working in Anhong medicine. And then today she is going to share with us the proteasome degrader as a new target for the new mechanism, for a new modality for drug development with cell specific. It's very difficult. So I think we will enjoy this talk.
Thanks, Margaret. Good afternoon, everyone, and thanks for staying so late for this last session. I'm so glad that I have the opportunity to be here, but I'm also a little bit skeptical because after all, I just joined Anhong in this January, so I'm pretty new in degrader. Not to say that this particular topic is actually not just on the degrader, but it's on antibody conjugated drugs. But when Margaret talked to me about this particular talk, I would like to know more about the protein degraders. I thought maybe we can actually combine these two talks together and then to come up with this very exciting new modality for cancer treatment in particular. But I must warn you that because Anhong is also just in the very beginning venturing into the degrader antibody area. So today, most of the data that I'll be using will be from different companies. But I would like to use their approach as an example because I believe that they lay out a very good roadmap for anyone who are interested in developing degrader antibody conjugate. So before we begin, I'd just like to emphasize that when we talk about ADC, I mean antibody drug conjugate. And then for protein degrader conjugation, I will use DAC instead. And so this is initial introduction. So let's take a look at the antibody drug conjugate. The concept was conceived back in 1993. So it was, I would say, a century-long journey. And even though the concept was conceived in 1993, but the first approved drug for ADC, it's approved for AML, was approved only in year 2000. So it's almost a century long. And unfortunately, even though the drug was approved, but it was actually withdrawn from the market 10 years later because of the low activity as well as the toxicity. And then the next ADC didn't come until 2018, 2019. And this is an antibody drug, conjugated drug that was designed to treat breast cancer. And after that, we begin to see a lot of drug antibody conjugate. And most of them are actually designed to treat cancer. So if we look at the structure of antibody drug conjugate, then you will have, of course, the antibody. Typically, it's a monoclonal antibody. And then because most of the ADC was designed to treat cancer, so most of the payload are actually cytotoxic drugs. And then the carrier and the payload are connected with linkers. And so once the ADC binds to the antigen, surface antigen on tumor cells, then this entire complex will be endocytosis into the lysosome. And then in the lysosome, antibody and the linker is clipped and then release the cytotoxic agent. So the cytotoxic agent can go about to do their things, mostly killing. And so this is a very simple concept. And the main advantage of ADC is that because you can use a very specific antibody, so you will be able to enhance the targeting. Otherwise, you might as well just use a common chemotherapy. And of course, with this kind of more specific targeting, definitely you will be able to reduce off-target effect. And also, because of the antibody drug conjugate, it will stay in the circulation longer, so it will prolong the circulation time and then minimize the resistance. But ever since the approval of the first antibody conjugated drugs, there are a lot of technical improvements. So for the first generation to the third generation ADC, we improved in the antibody to remove the mouse-derived antibody. We make the linker to be clippable. And then we also learned this technology to adjust the drug antibody ratio. And then still we are facing the problem that because some of the drug will be leaked into the bloodstream, and so we still have, for some drugs, have pretty narrow treatment window. And then, of course, if you cannot deliver sufficient drug into the cells, then the effectiveness will be lower. And even for the third generation antibody drugs conjugate, we still have some problem in replicating the conjugation technology. But more importantly, if we look at most of the payload, these are all cytotoxic agents, either targeting microtubule or DNA or RNA. And some tumors are intrinsically insensitive to microtubule binder. So even if you perfected all of this technology, you still have to deal with those cancer cells that are not sensitive to microtubule binders. So there's still room to improve and definitely room to imagine. So you can see the thousands of flavors of antibody drug conjugate or variety. So you can vary the carrier. You can also vary the payload. So you can see that the carrier have been evolved from antibody to peptide to small molecule or even viral-like particle or a pentamer. And definitely you can also modify the payload. So starting from the cytotoxic agent, you can also change it to radioligand as well as oligos. So you can conjugate siRNA. And definitely more creative way is to conjugate antibody to cells. And I'm gonna focus on the last one, the antibody degrader protein conjugate. So this is actually also one of the concepts that are recently evolved and has gaining a lot of interesting and exciting. So before we talk about degrader antibody conjugate, let's first talk about degraders. So protein degrader, currently most of the protein degraders utilize the ubiquitin proteasome system to degrade the target proteins. And so the UPS system is actually the endogenous machinery to maintain the endogenous protein homeostasis by degrading the unwanted or unneeded protein. So ubiquitin takes the unwanted or damaged protein which was recognized by E3 ligase. And then targeted proteins will be delivered to the proteasome. And proteasome actually behave like a shader, so break down everything into small peptides. And so with this kind of system, we can easily maintain all of the protein at the right level at the right time. So typically the UPS system, you will see the E3 ligase, which is responsible for recognizing the target protein. And then when you recognize the target protein and E3 ligase, as well as E2 ubiquitin form complex, then the ubiquitin will be transferred onto the target protein. And after multiple rounds of ubiquitination, the target protein become polyubiquitinated. It will be delivered to proteasome. Proteasome would degrade the target protein into small peptide, and the ubiquitin will be reused. So this system existed in every cell. We use this to maintain the protein homeostasis. And so the protein degrader system actually take advantage of the UPS system. So using this degradation machinery, but instead of letting it to target the regular endogenous protein, we try to create a drug we call it protein degrader, which will link the target protein to the E3 ligase. And so now, looking at the same system, you will have a disease-causing protein, and then your same E3 ligase complex, as well as your E2 ubiquitin complex. And initially, these two, the target disease-causing protein, may not be the native substrate for E3 ligase. So you create a drug, either a large molecule, well, it's a larger small molecule we call PROTAC, or you use a smaller drug, which we call molecular glue. And regardless of their design, the main function of this protein degrader is to bring these two into proximity. So once you have the degrader interacting with disease-causing protein and E3 ligase, it will bring it to proximity, and then the same system will kick in. So the disease-causing protein will be tagged by E2 ubiquitin system. And then once the system is formed, the drug dissociated from the complex, and the disease-causing protein is tagged by ubiquitin. And then later, the disease-causing protein is degraded, and the ubiquitin is reused. So this is an entire cycle, and the very simple concept of the protein degradation system that is used. So we harness the UPS for disease treatment. And here, I would like to draw your attention here. This is a very important concept. That is protein degrader, because the drug can dissociate from the complex, so the drug can be reused. So actually, an individual degrader can actually target multiple disease-causing protein. And this is very different. It actually has what we call catalytic recycling activity. And that makes the degrader much, much more potent than regular small molecular. Because for regular small molecular, you typically need to compete with the target protein for substrate for ligand binding. And so it's almost like 1-to-1. You need to have much higher drug concentration. So, so with this concept in mind, you probably think that, so it's a UPS system. You can only use this system to degrade the protein that's disease-causing protein. And so in cancer, it's not that rare. So most of the cancer or the disease that we've been thinking, also all of the targeted therapy, we pretty much design our drug against oncogene. So like EGFR or maybe androgen receptor. And so these are the target protein are elevated or oncogenic. It designed a degrader to directly degrade the target protein. But if you think about the mechanism, all of the endogenous protein are also degraded by certain degradation system. So for those target genes or target protein that are tumor suppressors or that are unregulated in the cancer, if we want to elevate the target protein level, you can still take advantage of the system. So instead of designing the degrader directly against the target protein, you can actually design a degrader to target the E3 ligase that cause the protein to degrade. So then you can stabilize your tumor suppressor. And so this is the case, for example, for p53, which is either depleted or downregulated or mutated in about 80% of cancer patients as suppressors. But it's degraded by MDM2, which is a ligase. So if you design a drug, for example, MD222, you can actually stabilize p53. So you can actually utilize this system for many different ways. And definitely, protein degrader has a lot of advantages. So as I mentioned before, catalytic recycling makes the drug very, very potent. And also because it's activity independent. When I say activity dependent, I'm talking about typically when we design inhibitors, our drug has to compete with the substrate or has to compete with the ligand. But when you design a degrader, it doesn't matter whether this target protein has any non-enzymatic function. And you can, as long as you can design a degrader target part of the structure of your target protein, and then you can achieve your goal. So you don't have to restrict yourself on enzymes or on receptors. And this actually opened up a very very wide possibility. So for a lot of drugs that are previously considered undruggable, now you can use this approach. And this also allows us to broaden our application. So it's a very promising drug type. But it's also very challenging because typically your degrader needs to interact with two proteins, your target protein, as well as your E3 ligase. So unlike your traditional inhibitors, it only need to consider drug and its target. So the design and also optimization is much, much more complex. And because the catalytic recycling activity, so the drug can definitely degrade your protein to extremely low level. And if your drug is not clean enough, so if it target also accidentally target other targets, then it may cause substantial toxicity. And then most of the PROTAC drug are much, much larger compared to regular small molecule. That makes the drug harder to get into the cells. And so it poses a lot of challenge in pharmacokinetics. And so when we look at this very promising but very challenging therapeutics, and then think about the advantage of antibody-conjugated drugs, maybe these two are match made in the heaven. So if you can combine the degrader with antibody, using the degrader as the payload of antibody drug conjugate, then you can derive the specificity from the antibody. And then you also take advantage of the potent activity of the degrader. So we hope to generate or to come up with a new type of therapeutics that has a better specificity with better potency. So this is the whole idea behind the degrader antibody conjugate. And then we can imagine that with this kind of design, the DACs will still bind to the cell surface antigen and then be endocytosis into a cell. And then in the endosome or lysosome, then the degrader will be released by clipping the linker. And then the degrader drug is released, so it goes by to degrade the target proteins. And then you can get the activity that you want. And because of the catalytic recycling activity, you can see, or you hope to see, a continued degradation of your protein interest and then cause cell death. So this seems like wonderful, but is it our fantasy? Or maybe we can take a look of the first degrader drug, antibody drug that's already entering into the clinical study. And so let me use the following two examples. GSPT1 degrader antibody conjugate as an example to show you how companies are thinking about designing this kind of drugs. And so before we talk about the drug, I will also introduce a company, which is Oron Therapeutics. This is a company in Korea, and this is the first biotech company specialized in degrader antibody conjugate. So we all know that targeted protein degrader has a lot of promise, but also is very challenging. And so they think that if we can conjugate the degrader to tissue or cell-specific antibody, we may be able to achieve a cell-specific degrader that would deliver specificity to the disease-causing cells. And so they actually begin their company by establishing a lot of technology. So first technology is that they use PROTAC technology that allows the PROTAC molecule, this is the degrader, to be easily conjugated to the antibody they select. And this actually will establish a platform that it doesn't matter which antibody and which drug that you want to conjugate, this will be pretty straightforward for them. And then once you establish that technology, they also begin to collect their antibody and their payloads. So they have a lot of degraders they generate, and they also have a good collection of antibodies. So by comparing the degraders and antibodies with proper pairing, you will be able to treat disease in many indications, and also not just the oncology, but a lot of neurological disease. And so with this technology, they actually build a very robust pipeline. So their DAC pipeline, including a lot of drugs using GSPT1 as a degrader, but with different antibody to target different cancers. And one of the drug, 5029, which I'm going to spend a little bit more time to explain to you how this drug is designed. And then also the same degrader, but conjugated to anti-CD33, was originally in the IND enabling stage, but already taken by BMS to treat AML. They also have other pipelines. And so this is the ORM 5029 is the first DACs entering into the clinical studies. So I believe most of you probably never heard of these target proteins. As an oncology, or if you're familiar with cancer therapy, then you're probably more familiar with oncogenes or tumor suppressor. But this is a very, very dead old housekeeping gene, GSPT1. It stands for G1 to S phase transition. And they actually selected this particular target for their DACs. And so if you look at the protein expression level, it's almost expressed in every tissue. And it's expressed in cytoplasm. And the protein level is actually quite equal in every cells, in every body. And this is a protein that is responsible to mediate the stop codon recognition. And so once it's recognized as stop codon, it's responsible to terminate the protein translation step. And if you disrupt or degrade this protein, it will lead to disrupted protein synthesis. And then ultimately, it will initiate a stress response and cause the cell to undergo apoptosis. So I call it target housekeeping gene for the kill. This is a completely different concept from when we just designed the degrader alone. Because we can use this kind of degrader and just attach any antibody so that we can use this to treat, say, like breast cancer versus lung cancer. And indeed, this is what they do. So they initially identified GSPT as their target, and they also have in-house small molecule membrane permeable, highly potent small molecule GSPT1 degrader. So with this degrader, they begin to think about which disease shall we target. So they take this degrader and screen more than 250 cell lines across 20 different tumor types. Even though we say that this is a housekeeping genes, but nonetheless, when you take a degrader targeting GSPT1 and compare the activity across different cancer types, they immediately see that GSPT1 degrader is quite effective for breast cancer, and particularly for those breast cancer cells that are HER2 positive. And the other group of cancer type that are quite susceptible to GSPT1 degrader is AML. So with that in mind, it's pretty easy. Now that you just need to conjugate a GSPT1 degrader to an antibody that will target HER2 expressing cells. So this is what they do. So they begin to construct the degrader antibody conjugate, and they end up with this particular drug we call ORM 5029. And when they compare the activity of this degrader antibody conjugate to just the drug itself or to other GSPT1 degrader that's already in clinical stage, they found that their degrader antibody conjugate is much, much more potent than all those other GSPT1 degrader. And when they also compare the degrader antibody conjugate to conventional standard therapy, they also found that it's much, much more effective compared to conventional therapy. And the drug antibody ratio is about 3.5. So they already show that the conjugation improved potency of this degrader. They also like to know when compared to other drugs or standard treatment across different cell lines with various level of HER2, would the drug still show pretty good activity? And as you can see, activity is quite good. So most of the cells, the activity is narromolar or subnarromolar for all of these HER2 positive cells, and much better than the antibody standard care or just the degrader alone. And of course, they know that this is a degrader. And we mentioned before, if the degrader is not clean enough, then the more potent is, it's likely also will be more toxic. So they conducted a proteomics analysis to treat cells with this degrader and look at which protein is degraded, and they come up with a single protein that was degraded. This is the target protein, GSPT1. And they also show that not only the drug can cause the degradation of GSPT1, it will also initiate the stress response. So as reflected by the expression level of 2 transcription level of ISR genes. So they proved that the degrader is doing what it's supposed to do, not just by, not only by degrading the target protein, but also initiate the downstream response. And then once you get down with in vitro activity, they also need to prove that the drug is efficacious in an in vivo model. So in an in vivo model, they compare the drug antibody conjugate with the degrader itself. So this is an unconjugated degrader. And you can see that the unconjugated degrader almost have no activity at all in the animal model because of the poor pharmacokinetics activity. Whereas the antibody show a pretty good activity, not just the high HER2 expressing cells, but also low HER2 expressing cells. It shows similar activity compared to the conventional, the standard care. And finally, they also show that the antibody can induce dose-dependent efficacy in the animal model, and also dose-dependent target protein degradation, as well as IRS response. So the antibody is doing, the drug degrader antibody conjugate is doing all the things it's supposed to do. And this is why they took this antibody, the drug antibody conjugate, into the clinical study. And so that's almost the end of the story. Not quite, because let's go back and look at the initial profiling study. We mentioned that they found that GSPT degrader is quite efficacious or quite active against breast cancer, particularly HER2 positive breast cancer. They also found that the GSPT1 degrader also is quite active against AML. So if we know anything about AML, these are leukemia cells. A lot of them actually have CD33 positive. And this is actually the first antibody drug conjugate is approved for using anti-CD33 antibody. And so with that same thinking, they actually took the same degrader molecule, and instead of attaching it to anti-HER2, they attach it to anti-CD3, and then trying to see whether you can get activity against AML. And as you can see that the antibody drug conjugate is actually very, very effective against MB411 cells. This is a CD33 positive AML. And compared to just the drug alone, or their drug small molecule, a drug that is designed for GSPT, but not conjugated with antibody. This drug is much, much more potent. And if you look at the IC50, typically you will see most of the IC50 will be in the picomolar range. And they also use the protein degradation assay to show that the drug antibody conjugate after 12 hours treatment can cause a complete degradation of GSPT1 in the target cells. And the unconjugated degraders, although they can cause degradation at higher concentration, but at lower concentration, you cannot see any activity. Again, this is due to the poor permeability. So they definitely will take this for clinical study. So then they also test the activity of this antibody drug conjugate. Now it has a different name because it's the same small molecule but conjugated to a different antibody. So the new drug is called ORM6151. And this is designed for AML cells. So they look at the 6151 activity against many different CD33 positive AML cell lines, and you can see that the activity is awesome. And but if you treat the CD33 negative cells, then the activity is much, much poor. So there are at least 100 to 1,000-fold difference between CD33 positive versus CD33 negative cells. So this actually also provide a good hint that the drug can specifically targeting cancer cells expressing high CD33, and it will be very safe for other cells like normal blood cells. And so before those data are cell line data, they also look at the cell line, actually human primary AML cells isolated from different patients. And these are all relapse or refractory patient. And you can see that, again, 6515 still show pretty good activity in these primary and refractory cells compared to standard care. And if you look at if they take this drug antibody conjugate to treat the healthy progenitor cells, they actually found that 6151 has very low activity. That means that this drug is very safe. It spared the normal healthy cells. And so this actually has a very, very big therapeutic window. And if we look at the unfortunate first antibody drug conjugate, Mylotarg, you will see that it's quite toxic against all of those progenitor cells. And this explains why this drug was approved from the market after 10 years on the market, or after 10 years after approval. So then the last test is that actually if you're familiar with AML, then the most difficult to treat patient will be those patients that are already entering into the blast stage. And so they actually isolated 6 different cells from patients who are in the blast stage. And so these are actually AML cells derived from 6 different patients. And again, you see a very promising activity in all 6 different blood cells. And so this is the last piece of data. They also show that in the in vivo activity assay, in the animal model, you can achieve pretty good tumor suppressing with a concentration as low as 0.1 mg per kg. And this is a single dose, single injection. So now you know why BMS took this drug, even before the drug entered into the phase 1. So this is my last slide. I think with all those data that we saw from Aurum, we know that when we pick a very potent and clean degrader and then attach it to a very specific or selective antibody, then we can definitely fulfill our promise that we can get a drug that has better specificity and better potency. But this is the first and the second drugs that are entering into the DAG area. And actually, with this kind of data, a lot of companies are getting very, very excited and trying to get into this area. But there's not that many companies actually have this expertise that can cover both the degrader as well as the antibody field. So you will see more and more companies or more and more collaborations between degraders company and antibody companies. And so this is, I think, a very promising future, and I hope that with this, you will be as excited as I am, and then perhaps we will have some opportunity to also start the effort for degrader antibody conjugate. Thanks.
Thanks for sharing your experience and also what your vision is. And so I think the audience, would you have some questions, Professor N?
Yes, for the HER2, anti-HER2 GSPT1 degrader, do you know which antibody they use? They use trastuzumab or?
Trastuzumab.
Yes, yeah.
The old drugs with the old antibody with new flavor. Yeah. And so actually in their data, they actually compared the antibody alone with the drug degrader antibody conjugate and show that the degrader antibody conjugate is about 100 to 1,000 times more potent than the antibody alone.
You said many people interested in this field. In addition to GSPT1, what's the next housekeeping gene people want to target?
What's next housekeeping genes? I haven't really looked into that many housekeeping genes because at home was mainly focusing on degraders design. So before, we mainly focusing on oncogenes or tumor suppressor genes. But so this is actually an eye-opener for me that instead of targeting what we think disease-relevant targets, you're actually using the degrader as a cytotoxic agent. So as long as the cells respond, you just use the antibody to take the drug or the cytotoxic agent to the cells. And I think the main advantage is that now you can actually think about other housekeeping genes that once disrupted, it will also cause cell death. And I think there are plenty, but I haven't really seen that many, at least entering into later stage.
Thank you.
Very good introduction. Thanks for a good talk. So in terms of DAC, it's pretty much like targeted pro-DAC.
Yes.
Right. So I'm wondering that in terms of selecting the antibody target as well as the targeted protein to be degraded, is there any association between them? That, for example, like say, if there's some targeted protein have close correlation to the target, antibody target, that might be better choice?
Actually, I'm not really sure, because when, if you look at their approach, they already define on the GSPT1, but they also need to go through a lot of screening to identify potential disease. So quite likely that if, in a way, maybe antibody dependent or target protein dependent. And if you look at GSPT1 in the pan-cancer study, only less than 1% of cancer patient has any alteration in GSPT1. So if you try to go from those previous data and trying to identify the association between disease, cancer type and the drug target, it will be difficult to find. So I believe this is why they actually take this type of approach.
Yeah, I'm just want to kind of get your take on this one. That, for example, like say, HER2 expression that's highly upregulated in breast cancer or other kind of cancer type, but it's also impact on downstream signaling, right? In terms of downstream signaling, that there some other protein upregulated as well. So if you say you target the HER2, at the same time it degraded the downstream signaling protein, wouldn't that be kind of kill two birds in one stone?
Oh, definitely, definitely. Yeah, but if that's the approach, then you do need to do a lot of tests beforehand. So you need to know the linkage between your specific antibody or the antigen and your target proteins. And if we talk about housekeeping genes, then you probably just can only imagine, for example, those oncogenes activated cells that go through a lot of cell cycling and then perhaps have elevated protein synthesis activity or RNA synthesis activity, and you hit those target, and then you also look for antigen or oncogenes that cause that kind of phenotype. Otherwise, when I saw this approach, I went into CBio portal and trying to see if I can find any positive or negative correlation between, for example, HER2 or CD33 and GSPT1. And since this protein hardly changed in different cancer types, so it's actually quite difficult to go from just a wet lab, just from the dry lab approach.
Can you share a little bit experience about the design of the degrader? How big or what about the range of it? And I think I heard that your company is using AI or brain power to design the degrader. So can you just share a little bit of the secret?
Yeah, actually there are two types of degrader design. So the original type of degrader design we call PROTECT. And let me see if I have, yeah, here. So PROTECT molecule typically have two ends. One end recognize E3 ligase, the other end recognize your protein of interest. And then these two domains, I'm talking about small chemical structure, were linked by a linker. So it's very similar to antibody drug conjugate, except both ends are chemical structure. So PROTECT typically has a molecular weight greater than 800, which is not that big compared to antibody. But when you try to have this kind of drug entering into cells, it's a big problem. And then also another type of degrader, which is a smaller molecule. So instead of having a linker, the region that recognize E3 ligase and the region that recognize protein of interest are actually very, very close. So it will be much smaller. And so we call that molecular glue. So that molecule actually bind to the interface between E3 ligase and your target protein. And so typically, you may not need to use antibody to bring the molecular glue into the cells. But molecular glue is much, much harder to design through a rational design. Actually, most of the molecular glue before were discovered by phenotypes screening. So you see the activity, and after, say, like 10, 20 years, for example, thalidomide, then you know that it's a molecular glue that hit one side at the E3 ligase, the other side at the target protein. So most of the company who would think about using the antibody to bring the drug in will first consider the PROTAC molecule. PROTAC will be easier to design using AI approach because you can use computer to model the structure of your E3 ligase as well as the structure of your target protein. And then you use generative AI to generate potential chemical structure that can bind to your E3 ligase and can bind to your target protein. Then you use another algorithm to link these two pieces together. And then you take the virtual compound and then go through a virtual screening. And from there, you can identify promising chemical structure. Then you move from dry lab to wet lab to synthesize those chemicals and then test that in a cell-based system.
Yeah. So anyway, we really appreciate you actually not only introducing your degrader chemistry and PROTAC concept, and also even top up, think about how to connect that with the antibody so that bring us more opportunity. We hope that next year we can hear more successful story about all the advances. And really, I think we should close all this section with a highlight of today's talk. Thank you very much.
Thank you.
Thanks again for the wonderful two talks and the moderator, Dr. Zhang. Okay, yes, we are approaching to the end of today's conference. So please welcome Dr. Hanzhong Wu to deliver the closing remarks. Please welcome Dr. Wu.
Yeah, I think I stand here. Okay. Yeah, greeting to our distinguished guests in person and online. So today's conference is come to the conclusion. So I would like to take this opportunity to thank all of our distinguished speakers that gave us a wonderful talk and bring us a very innovative technology. As we know from the new drug development for treatment of thyroid disease, cancer therapy, as well as other disease. So the other has also talked about antibody engineering, like use the increasing affinity or therapeutic efficacy. I also use the ENAP to recognize different kind of epitope and to produce the power top antibody. It's quite effective, increase the therapeutic efficacy and antibody avidity. So also later this afternoon section also talk about antibody-based therapeutic, we so-called a new modality. Okay, it's including the bispecific antibody, is delivered by Dr. Ho, and also have antibody drug conjugate. Okay, yes, quite very efficacy, increase the efficacy. Also have the ATC, also the DAC. Yes, PROTAC. Yeah, also have with a good potential for treatment cancer. The other is Dr. who also has given us a very interesting talk. Is use the AAV for gene delivery for the gene therapy. So also has get a very amazing clinical data. It's approved by the EMA and recently also quite approved by the FDA. Yeah. So I think we thanks all of these distinguished speaker, and let's give a big hand to this speaker. The other, I also would like to thank our many organizer and the co-organizer and the sponsor just listed here. Without their support, cannot make these events possible. Also, I also would like to thank our colleague from the BioTrack, Demi Sinica. He has given us a big support, okay, in making this conference successful. So here I also want to announce in tomorrow, we also invited 5 excellent, outstanding researchers to give us a talk. He has a talk about his very novel, innovative research, as well as his research career. And we are also very honored to invite the Professor Ann from the UT Health. He will deliver give us 3 hours workshop. He's focused on antibody drug discovery and the development. So don't miss in this very inspiring and stimulating talk. Okay, so I think I also look forward. We have more collaboration from our researcher, particularly with academic and the industry, that us we all can continue to innovate, to inspiring change, as well as collaboration to increase, to promote human health and the welfare. Okay, thank you.
Sorry, we will have the group photo. We invite all speakers today and the moderator to come onto the stage to have a group photo. Thank you. And meanwhile, please remember tomorrow's registration time is 8:30, and the event will start at 9 o'clock in the morning. Thank you. 今天的讲者、主持人，请上台一起合照好吗？谢谢。